Yeah, hello everyone. Thank you so much for joining. And yeah, I'm Laura. I'm going to talk about how scarabs interact with infrared light. And so it is really exciting to study living light. But today I will be talking about a specific kind of light, infrared, which refers to longer wavelengths. In this talk, I will talk about a range between 700 and 1700 nanometers. Why is it important to study this light? Because from all the light that comes coming from the sun, only 45% of the energy is in the UVB range. So if we focus only in that range, we're missing out on half of the fun. For example, reflecting near infrared has been proposed as a way to get rid of all that extra radiation. Not long ago, researchers created this smart fabric that can filter or trap near infrared to aid in human thermoregulation. This may, however, look like child's play for the Saharan ants that for millions of years have been covered in silver hairs that reflect infrared light to avoid overheating. So it is useful to understand how animals interact with this light, mainly for thermoregulation, and maybe apply this knowledge into our own technology. But what's the problem? That mechanisms for interacting with near infrared are relatively unexplored. So I want you to look at this set of scarabs, but under infrared light glasses. We don't really care about the amount of infrared that is being reflected, but it's not only black and white. For example, these three reflect a lot of infrared, but they are slightly different, right? Thus, my question is, do scarabs have different mechanisms to interact with infrared light? We call these interactions signatures because they are not limited to how much they reflect. They can be a very unique collection of optical effects, determined by the size, shape, and disorder in the structure. Let me introduce you to the superstars of this talk, these three scarabs, and their signatures. Yes, they all have high infrared reflectance, but the first one has actually a blue shift with the increase in illumination angle. So this one is the iridescent one. The second one has a broad band and diffuse reflectance, and this one is similar but has outstandingly low transmittance, the third one. So for now, we're just going to color code them like this to remember. And our hypothesis is that the iridescent one needs to have an ordered structure, and the other two would have a more disordered structure. And we are testing this in three simple steps. The first one is to have a look at electron microscopy photographs of the beetle shell, as here, and based on optics theory, we propose that the structure highlighted in red are the ones interacting with infrared. But that is not enough, right? We need to test that they actually meet the requirements to produce that specific near-infrared signatures. And to do that, we obtain many TEM images of these structures and quantify some parameters. I'm showing you the most important ones. The thickness of these pseudo layers here, and in this case, the size of these air holes, their separation, and how ordered they are. And here, maybe how much empty space it's there. So we, after analyzing these images, we can kind of simplify the structures so we can actually fit those things in our models, those simple structures. For the first beetle, we propose this mechanism of fibers rotating to form a helicoidal structure. This is well known to occur in scarabs, but has only been studied for visible wavelengths. For the second beetle, we propose a matrix of kiting containing air cylinders. And finally, for the third one, we propose a very disordered array with even bigger hollow spaces. So now let's test if it interacts with infrared. First, we convert this complex 3D array into a 2D structure that is illuminated from above, and our first approach is to assume that this distance in the rotating layers is constant. And this structure actually produces a blue shift with the increasing angle, which is the iridescence. But this we only did for the visible light. So now let's see what happens if we illuminate it with light of different wavelengths. On the left, the lighter regions indicate the depth at which this wavelength is interacting with the structure. And on the right, you will see the overall amount of light reflected. So the longer the wavelength, the deeper it penetrates in the structure, but this configuration produces one sharp peak in the visible. So not there yet, right? But what we're trying to do is to test if the distance in the deeper layers is larger and optimize to interact with longer wavelengths. With this change, the total reflection of longer wavelength increases. So now we want to combine this configuration with what I showed you before to test if we can replicate the iridescent effect that we measured with the spectrometer. And who knows, maybe see if there is some infrared polarization. Now our second beetle is super interesting because its structure is in the middle between order and disorder. So we chose to compare these two models. In the first one, there are cylinders of varying sizes, but they are all aligned, and this allows some differences in the interaction with light depending on its polarization. And overall, it would reflect infrared light, but it seems that it will also reflect a lot of visible light. So maybe we can consider this other model that incorporates a bit more disorder. That has been studied that have been used to study the diffuse reflection of blue light in birds. And so we adapted this code to calculate for each wavelength the probability of it being reflected by the structure. And the results indicate that longer wavelengths have indeed higher probability of being reflected. These two are negative controls that we also run of structures that we don't expect to interact with longer wavelengths in this way. So it seems that the broadband diffuse profile of this beetle may be given by a quasi-order structure. And for this last beetle, we think that light enters in this cavernous space and bounces everywhere, and some of it will be reflected maybe by incoherent scattering, such as it happens in other beetle mechanisms, but some of it will be also trapped. And it's interesting because there's this material that has been developed as a great insulator for both heat and also light, and it looks exactly like this beetle. So, I mean, this structure is not tuned to reflect only one wavelength, but maybe if it reflects a high proportion of light and whatever is not reflected is trapped, it could be good for thermoregulation. So we're working in a model to test this theory. But for now, we can wrap up with a bit of a take-home message. Sometimes we may feel tempted to assume that it only takes bigger structures to interact with longer wavelengths, or that all organisms reflect infrared in the same way. But that does not seem to be the case. We discovered that these three scarabs produce three different infrared signatures, and that is only possible because they have different structures with specific geometries and characteristics that determine what they can do to light. Here we showed that actually in three scarabs that we studied, three different mechanisms were found. So the answer to our question seems to be yes, they do have different ways to interact with light. So what? Why do we care about this diversity? Because it opens these kind of questions. Are all of them equally effective for thermoregulation? Are they equally easy to produce? Can they be combined to obtain different optical effects? And can they inspire new technologies? So stay tuned because we're working on some of these. And finally, I just want to thank my team, my PhD supervisors and collaborators, lab mates, and friends who make this research possible. But above all, I want to thank you so much for attending to my talk. And I would be delighted to receive all your questions, feedbacks, ideas in person, in the chat, or even in social media. Thank you so much.
Very nice talk. I didn't see any question in the chat, but I do have a question. So it's a technical thing. When you're modeling the near IR, so sometimes near IR refractive index, how did you get those data? Sometimes it's difficult to get those data. Did you measure them or you just estimate?
the index. Yeah, we
Yes.
had to estimate it because definitely is difficult. So yeah, we're working in kind of like a margin of error. We're just estimating some things and then running the model and seeing what happens in the most extreme scenarios, because definitely measuring is kind of hard. Yeah.
Cool. Yeah. Thank you. Yeah. So
Thank you.
next speaker will be Kiwi. Should I?
Yeah. Let me share my screen.
Yeah. While you are sharing, let me mention one thing. So we are recording all the way through. So I know only a few of you didn't agree to be recorded. So we know that we will delete those recording because this is easier for us to manage. Yeah. So yeah, Kiwi, yeah, you can start.
Okay. Right. Hi, everyone. Thanks for coming to my talk, where I'm going to present my work on the morphogenesis of nanostructures in butterfly scales. So far, in the conference, we have seen many of the vibrant colors and patterns in nature, particularly in invertebrates. So, for example, insects. So, insects are one of the popular research subjects because of their diversity and also relative ease of manipulation. For myself, my focus is on the butterflies, particularly on their wing coloration. So these colors are produced by tiny cells on the wings known as scales. And these scale cells, they are arranged on the wing like roof tiles. Each scale cell, each single cell, produces one color, which can be pigmentary or structure colored or a combination of both. So each scale cell is held to the wing by socket cells, which is these bits over here. And this is a small zoom-in area on one single scale from a top-down view. This is a cross-sectional view of a single scale as well. So a scale cell typically has two surfaces, the upper lamina, which has long ridges that are connected by cross ribs, and the lower lamina, which is generally unstructured. Both surfaces are connected by what we call trabeculae, which are these pillar-like structures. And together they enclose this space, which we call a lumen. Each of these structures can be modified to produce different colors. So here are a few examples of modified ridges, cross ribs, and also the thickness of the lower lamina can affect the blue coloration of the butterfly, and also some gyroid crystals within the lumen can produce green coloration. So far, research in butterfly wings are heavily focused on identifying the adult structures and the optics that's involved in producing the structure colors. But not much is known about how these structures are formed, which is the research question that I'm asking in my study. So one thing we know about butterfly scales is that they share the same developmental pathway as bristles in Drosophila. So in Drosophila, there is a shaft and a socket cell, whereas in butterflies, there are scales and socket cells. Previous studies have identified that the position of the ridges on the surface is predetermined by position of actin bundles within the cell. So during cell growth, actin bundles are positioned underneath the plasma membrane. So you can see these bits over here and also the lower surface as well. And the ridges are formed in between adjacent actin bundles. As the cell continues growing, the cells become wider and flatter, and the ridges becomes more and more prominent. So these findings have been validated in several studies, but including using other techniques like immunofluorescence. As you can see over here in magenta, in purple, it's WGA, region agglutinin, which stands for chitin, which is part of the cuticle. And also in yellow is phalloidin that stands for actin. And again, you can see that in the cross-section, the ridges is always forming in between the actin bundles. As the cell matures and dies, the actin is degraded and the chitinous structure is left behind. So adult scales are essentially dead cells. So these findings are validated in several studies, but they are generally limited to moths and the nymphalic butterflies. So currently, there's no data published on one of the largest families, which is the papilioniid butterflies, which is also an important question because papilionids have the most diverse types of wing scale nanostructures among the Lepidopterus. So in particular, many of the papilionid species have these irregular cross ribs, which are different from the typical nymphalid scales. So by comparing the development of species from both families, these findings can potentially give us some insights on how different scale structures are formed. The species used in my study is Paridis eurymedis, which is one of the papilionid species. So these species have irregular cross ribs and can see here. And they also have these thick walls that extend into the lumen of the scale. We call it the honeycomb lattice because the cross ribs resemble the honeycomb pattern. And for my study, I use immunofluorescence to follow the formation of these honeycomb lattice structures. So first of all, early developmental stages of scale growth are conserved. So these are pupal wings that are 8 days after pupation. And the corresponding developmental percentage over here. So in green, again, is the WGA that stands for chitin. And in red are the actin filaments, which at this stage, they are mostly clustered in the middle of the cell. So the actin bundles become thicker as the cells continue to grow. At the same time, they become more spaced out. And again, ridges are forming in between the actin bundles. But instead of disintegrating and disappearing after ridge formation, the actin bundles in red over here reorganize around these disc-like structures that we are also seeing for the first time. So these green disc structures, we call it the chitin disc. They are based on their location and so their morphological similarities to the adult cross rib patterns. These are the future cross ribs. So over here, I have observations from different angles. This is the top-down view. This is a zoom in of one of the areas, and this is the cross-section over here. And the association of actin, these reorganized actin structures with the chitin disc is the clearest when viewed in 3D. So over these small insets, which corresponds to the area over here. So a single chitin disc. So you can see as the cell continues to grow, the reorganized actin structures become more and more associated with the disc until the disc eventually become hollow. So it's much like the final morphology. And I actually have some videos here of the 3D stacks, the Z stacks to illustrate my point. So I'm gonna fast forward a bit. So you can see here are the double rows of the chitin disc, and you have the actin that is surrounding the disc. And 2 days later, for this species, which is about 67% development. So there's a lot of association of the red actin structures with the chitin disc. And also these actin structures extend into the lumen of the scale. And over here, which is towards the end of the development, you can see that they are now hollow. And again, actin is surrounding these structures. So this is the first time we see such non-linear actin structures in butterfly scales, but this type of structures have actually been observed in diatoms. So diatoms have cell walls that are made out of silica. And it was found that actin also forms these irregular structures during cell wall formation, which are proposed to be involved in templating silica deposition. So besides the reorganized actin structures, I mentioned that we're also seeing the chitin disc for the first time. So to find out how these discs they are formed, I use a combination of chitin and plasma membrane stains this time, because chitin, which is cuticle, is always deposited on top of the plasma membrane. So in just 1 day, plasma membrane in red over here went from relatively featureless to having these kind of irregular cross-rib patterns here. And in the cross section, you can see that chitin is still sitting on the surface of the cells as one uniform layer over here. So within the next 24 hours or so, cuticle accumulates into these spaces. And there are now a type of chitin disc structures that are sitting on top of the membrane. So this means that plasma membrane, they form a template or a scaffold, and cuticle is deposited into this template to form the disc-like structures. So these disc-like structures are also seen in several other papillonid species. There also are some hollow disc types in these species as well. And although there are some differences, these observations support that the chitin disc structures, they are conserved during the development of certain papillonid species. So this is the schematic of the process of honeycomb lattice formation that I'm proposing. So in early stages, actin bundles prefigure the position.
Sorry to interrupt. I think, yeah, 1 minute left. Yeah, I think this is the last, right?
Sure. Yeah, this is my last slide. So after the formation of ridges, actin reorganizes, and this forms because of the plasma membrane template. And this draws in the actin, somehow draws in the plasma membrane. And because chitin is deposited on top of the plasma membrane, it follows the shape of the plasma membrane to form these honeycomb structures. So yeah, this is my work summarized in 10 or 12 minutes. And if you're interested to know more, my PhD supervisor Vinod and myself, we are happy to share more details and discuss further. So just feel free to drop us an email or a text, and we can talk more there. Yeah, thank you.
Cool. Thank you. Very nice image and videos. And next speaker will be Kenza. Is here, right? And after Kenza is Maria Sol Vidal. I think. Okay, you're here. Great. Now you can start.
Okay, so I'm Kenza Djeghdi, and I'm going to be talking about 3D tomographic analysis, and the samples are diamond-based structure that were taken from weevils. So the thing about structural colors that I'm interested in is the type of structures that give rise to them. Just going to take the laser. So you can see a couple of examples here, and these structures, they include simple multi-layer stack like here, all the way to more complex 3D structure here, like this gyrate. So for ordered structure, the mechanisms are known, and we know why we have coloration, but what's also interesting, and it's been observed, is that some disordered structure, like those two, they also give rise to color, and this is less understood. So it's often going to be blue coloration. But if you have a structure that is too disordered, like this one, then you get something that is white because of multiple scattering. So from this slide, you can already see two challenges. Is that for one, we don't know what too disordered means because there's no real way to quantify disorder. And the second problem is that those are all 2D images. So for a multi-layer stack, this is fine. But for a gyrate structure already, since it's 3D, you have to take my word for it that this is a gyrate. And for disordered structures like those ones, it's almost impossible to know which kind of structure you're looking at just from looking at one 2D picture. So we decided to go and take 3D data. So there are many ways to take 3D data. In our length scale of interest, it's mostly going to be between those three techniques. The issue with X-ray tomography, it's been done and it works, but you require access to a synchrotron. And the issue with TM tomography, like here, is that the sample preparation is quite heavy and you get access to a very limited volume. So what we decided to do was to use FIB-SEM tomography, and you can see that in terms of volume that you can access and in terms of the voxel resolution, it's right in between those two techniques, and that's exactly what we needed. So we're lucky enough to have a FIB in-house. The way this machine works is that it has two beams, and they're at an angle to each other. So the electron beam here is just here to take images, and the ion beam at an angle here is the one that's going to be milling away the slices. So this is going to work. So every time the FIB takes the slices away, then SEM is going to take a picture, and you alternate those two over and over again until you get an image stack. So they're all 2D, and when you put them together, you can reconstruct a 3D volume. So if you do this for about 6 hours, and that's something we usually run overnight, then you get a volume that is up to 5 by 5 by 5 micrometer cube, and you get slices that can go down to 10 nanometer. So that's also going to be your resolution. So we decided to use this technique on this weevil here, especially its scales. So you can see here that it has two types of scales. And the red one are faceted. So you can see it more clearly here in the reflection spectra. There are 3 peaks, and also here in the K-space image. The blue scale, however, they're much more homogeneous. They don't have facets. You can see it here too. And their peaks here in the reflection spectra are less intense, broader, and have a higher background. So that means that those ones have scattering, and that's a clue that that might be disordered into this one, where those ones should be ordered. So to make sure, we have a first look inside with electron microscopy, and at this point, we're still just looking at 2D. And we can already see that they are widely different in order. So this one has clear hexagonal pattern and periodic features in all directions, whereas the other one is highly tortuous and looks random throughout. And this is also very clear in the FFTs here. So at this point, we saw that getting 3D data would be challenging because since we have a porous medium of chitin in air, we have a depth of field. And that means that it's really hard to get 3D reconstruction from this because we see all the next layers. So we needed to fill the structure with something. And we first tried to fill it with the silicon resin here, which is quite a standard protocol, but this was a very hard process to control, and the quality of the images we got was really poor because of the lack of conductivity of the resin. So we quickly moved away from this, and we decided to try an electron beam induced deposition. So this is a method that you can do directly in the field chamber, and that's going to allow you to deposit platinum in the air phase. So directly in the chamber from a gaseous precursor. So this has great advantages over the other method because it's in situ, and that gives you a precise control over both the location and the depth of the penetration. It also gives you great contrast since it's so conductive. So here the platinum is the white phase, and it protects your structure. So that's a win on all sides. So once you do this filling and you do the tomography, you're left with a couple hundred images. So the first thing we need to do is to realign everything to compensate for the tilts that was induced and the drift that was in the tilt. The second thing we do is that we crop a region of interest and we binarize it. So we give a value of 0 or 1 to each of the face. And that is made much more easy by the fact that we fill it with platinum. And once you've done all of this, you have a volume that you can reconstruct. So this volume here is quite big, and that's a good compromise between what we need in terms of information and what we have time to do and what the 3D software can take. So we do this for both types of scales, and those are going to be color-coded for the red and blue scale. And once you do this, you can take cross-section out of those structures. So that's much better than a TM because instead of having one slice, you can take cross-section from any direction. And once we do this, we can compare it to photonic crystals that are known to be found in nature. So here, in the interest of time, I'm just going to present the diamond, which I plotted using this level set. And for highly ordered symmetric structures like this one, you know exactly which pattern you're going to get depending on the direction that you cut it. And here you can see that I extracted a number of cross-sections from the diamond, and I could find always a match with the red scale. So that tells us that the red scale is almost a perfect diamond. The blue scale, however, regardless of the direction of cutting, it always looks completely random. So we have two volumes here that look objectively different, but we need a way to quantify that. So the way to go about it is to do first a step called skeletonization. So that's going to be the green part here on the right. And this is just a way to limit every connection between nodes to a single pixel line. So that's what we get. So why do we do this? We do this because that's going to give us access to quantification of some parameters, like here, the strut length, the bond angle, the dihedral angle, the connectivity. So here, without going into details, if you look at the distribution of all those parameters between the red and the blue scales, what you see is that the distribution shape is quite similar in every case. The only difference really is that there's a wider spread in the blue scale. So that tells us that very locally, the structure looks quite similar and that the blue scale is not completely amorphous because those values are not randomly distributed. We see here for the dihedral angle that we have much more of a spread, and that's a measure at medium range. So it tells us that maybe that's where the difference occur in order. So we decided to look at different length scale to compare the correlation, and we see that very locally, both of the structure have tetrahedral units, a majority of them. The difference is that for the red scale here, they are arranged at fixed angle, and that's what makes it look so ordered. Whereas for the blue scale, you have random torsion angles between the units, and that's what makes it look so tortuous. And when you zoom out even more, you can see clearly the difference. So if you want an idea of the length over which order gives way to disorder, you can look at the radial distribution function here. And you see that for the red scale, you have very narrow, intense, numerous peaks up to the 6 neighbors. Whereas for the blue scale, you have the first 2 peaks, which is a sign of local order, but then it gets drowned out in the background, which is a sign that you're lacking long-range order. Last thing that is worth mentioning, but I won't have time to go into details, is that you can feed those tomography directly in an FDTD software, and that's going to give you an understanding of the optics behind it. So you can simulate the reflection spectra and compare with experiment. But what we're interested in is this part where you can simulate the density of states in the structure, and that's going to give you an understanding of whether or not a band gap is opening. And that's the big question: is why does a band gap open in disordered structures? So we're reaching the end of this talk, and as an overview, I just showed you a tomography protocol that allows us to reconstruct ordered and disordered structures and leads us to quantification and qualitative analysis of those. So our next step would be to extend this method to other structures like layer structure by continuous packing, and to find a way to quantify disorder and find a common criteria that gives rise to coloration in disordered media. So I'd like to thank the Adolf Merkel Institute, the Soft Matter Physics Group, and in particular the Biophotonic subgroup here, as well as all our funding organisms, and all of you for attention. And I can be contacted here at this address if you have any questions.
Yeah, here is a question from Vino. He says, very nice talk. And what is the coordination number in the blue scale amorphous nanostructure? Does it vary between 3 and 4? Or is it
yes.
still many fourfold connected by the disorder?
So for both of the structure, the connectivity is between 3 and 4, closer to 4. So for the red one, it's at 3.8, which is really close to the 4 coordination weight that we expect for a diamond. And for the blue, it's at 3.5. But the angles are very close to tetrahedral angle. It's at 111 degrees. But we have a little bit of a spread.
Cool. Yeah. So let's move to the next speaker, which is Maria Sol Vidal. And after Maria, so Janis Heuer is here. Okay, Maria, you can start. Okay, yeah.
Hello, everyone. My name is Maria Sol Vidal, and today I'm going to talk to you about my work that I have done under the supervision of Dr. Diana Skigin at the University of Buenos Aires. Our work is titled Rayleigh Method for the Study of the Optical Response of Natural Interfaces. So the main purpose of our work was to develop a computational tool based on what it's called the Rayleigh method in order to solve the problem of electromagnetic scattering by a rough interface that can have an arbitrary function as a profile. And this is very important in order to study biological structures that in general they have a really complex profile. So mainly this method is based on what it's called the Rayleigh hypothesis, that it states that the scattered fields on both sides of the curvature can be described as a summation of plane waves that travels away from the surface. And it's important to say that this is a rigorous method that it's based on the exact resolution of Maxwell's equation with its respective boundary conditions. So the first thing that we have done is to implement numerically this method, and with that we can compute the reflectance and transmittance spectra for different lightning conditions, such as the type of incident beam, that it can be a plane wave or a Gaussian beam, the angle of incidence, the wavelength of the light, and the polarization mode. And after we have numerically implement the method, we have validate our implementation with several checkups. The first one we have made is numerical checkups, such as the stability and convergence of the solution. Then we have made more physical checkups, such as to verify if the energy balance criterion is satisfied, that is to say, the conservation of energy. We have also checked that the boundary conditions of the Maxwell equations are also satisfied. And lastly, we have compared our own results with known results from, for example, physical optics, and then with also previously published results in different scientific articles that I have listed here. And in all cases, we have obtained a really satisfactory agreement between the results obtained with our method and the other rigorous method, for example, from these papers or from the results from physical optics. And the novelty of our work is that we develop a formalism in order to incorporate the profile of the scattering structure into our code by means of a digital image of it. So here what you can see is a TEM image of an eugulena that then I am going to talk to you about this. And the main goal is to obtain the coordinates of this profile in order to use it in our Rayleigh method. And this formalism, it's mainly based on two processing stages, one with image shade for pre-processing and in order to obtain the X and Y coordinates with the tracing tool. And the second, it was with Python in order to adjust the coordinates that we have obtained previously to the requirements of the theoretical formalism that we have developed. And now what I am going to talk to you about are two examples of the logical structures that we have applied our method. The first one is for a TEM image of an Eugulena gracilis that maybe these are unicellular organisms that are really interesting because they can survive to ultraviolet radiation. So here above you can see the original TEM image, the original profile. Then here what you can see is the profile that we have obtained with our method. And then one of the advantages of working with this formalist that we are working with the exact profile of the structure is that, for example, we can study the dependence of the reflectance or transmittance on the irregularities of the structure. And one form of doing this is, for example, to study the electromagnetic response of this irregular structure and compare it with some kind of regular structure that was made from this irregular structure, averaging the different protuberances of this irregular structure. So the idea is to compare the electromagnetic response of both of these and see if they are different. So this is what we have done. In the left, you can see the reflectance of the irregular structure for a specific choice of parameters. And in the right, you can see the reflectance of the regular structure. And in order to validate the results of our method, we can compare it with the reflectance obtained with another method called Photonic simulation method that it's completely different from ours. And this method can be found in this paper that I write here. And it is inspired by the propagation of mechanical waves along a two-dimensional array of elastically coupled particles in a plane. And it's really different from ours. And now you can see that we have obtained a really good agreement between our results and this method. So this was also another way to validate that our formalism was working right. And the second example I am going to show you is about the Dione vanillae butterfly, that it has really highly reflective areas on its wings, on the scales of its wings. And the idea behind this is that it's structural color. So here again, you can see a TEM image of the cross section of a scale of this butterfly. It's already processed. And with the formalism we developed, we were able to extract the upper profile that you can see here. But here, the limitation of our code is that we are able only to take one interface. And as you can see here, this structure is made up by 3 layers, but actually currently in our formalism, we are able only to take one single interface. So what we have done with this, in order to illustrate only the power of our method, is, for example, that we can also study the dependence of the reflectance with the wavelength of the incident light. And for that, for a specific wavelength, what we do is to identify the main peaks corresponding to some orders, but then we have to calculate the total power that order takes, and this is done by integration, because we are dealing with finite structures. So here what I am showing you is the power, the reflected power versus the incident wavelength for example, different angles, and for the S mode in the left and the P mode in the right. And the idea is that this is really important when studying natural structures, and we can do this with this tool that we have developed. Future work that we haven't done yet is develop our model for multi-layer structure with more than one single interface. Also, to add to the code the possibility of anisotropic media, not isotropic, and also to incorporate structures that haven't a translational symmetry in one of the axes. And that's it, and thank you very much for your attention.
Cool, nice talk. Does anyone has any question?
I have a question.
Yes.
When you showed the structure that had 3 layers, they were little like little
yes.
rounded edges to the peaks of each diffraction grating peak. Are those included in the model as well?
No, because I haven't mentioned this, but the method requires that the function, it has to be single value. And that is to mean that it has to be a function. And these little circles doesn't satisfy that condition. So in that way, we have to average in order that, to obtain a single value function.
Okay, thanks.
Thanks.
It seems I was connected just now. So, so we next is I think before Janis there will be a empty spot because somebody dropped off. So let's take a coffee break, and we can also ask a question for the previous speakers, and then we will come back after, let's see, 14 minutes. So London time 6 a.m. So will be Janis. I saw you, Janis, here. I guess probably some of you, if you attended the previous session, some of you maybe felt tired running the session maybe on the same day. So Laura, if possible, can I ask you a question for the
Yep.
infrared? So at this point, you are looking at just near IR, am I right? You're not
Yeah.
looking at the middle IR, which may also play an important role in the thermal regulation.
Oh, true. We're mostly looking at only until up to 1700. Because definitely beyond that, it would affect like the emissivity could affect like the thermal regulation. But at the moment, we're testing more whether the properties of the LITRA can reflect the the reflection of these relatively short wavelengths, including visible and near infrared, whether that can be still kind of a buffer in challenging environments. And so, yeah, these Christmas beetles, they live in super hot environments here in Australia, in temperatures up to 40, 40 degrees. So, and they are active during summer and during the day. So it's really, really, it would be really cool to see if they actually have these properties.
Okay. Did you gather the live species to Singapore? I believe I remember you are in Singapore, right?
Oh, I'm in Melbourne.
Oh my bad. Okay, so
That's all right.
Okay, so you got this live species, am I right?
Oh, so
Live
for
animals.
this, yeah, for this first part, we did most of it with like museum specimens, because it's like as in it's better to have like the full like sampling the full diversity, and also I don't have to kill them. so yeah, with most of it with museum specimens, and what I'm doing for the thermoregulation stuff is we measure the thermal properties of the elytra only, or maybe the recently dead beetles. So some of them, they when they are out in summer, we can collect some species, but just a few. So then it's difficult to compare. But still, yeah, we collect some, and then we keep them alive, and then when they die, we do the experiments. Yeah.
Cool. Thanks.
Thanks.
So you guys feel free talking. I don't want to be the only person talking, asking questions.
So were a lot of you able to come to the first session?
Yeah, I was here in the first session. It was very interesting. It was funny because it was late here in Melbourne. I confess I was in my PJs, but it was fun. It's really good. Yeah, so many interesting things.
Yeah, it was perfect time in Europe yesterday, but no, I had to wake up at 5 today.
Oh God.
The sun is still not up.
Oh, where are you?
I'm in Switzerland.
Oh, okay. Oh.
Yeah, the lighting is terrible because my lamp died and there's no sun. So I'm basically in the dark.
Yeah, the challenges of the online.
I think something for the rocks.
Yeah, well, yeah, it's 10 to 6 here.
Yeah.
Did you guys get sleep?
I tried to
was
go
too excited
to bed
to sleep.
earlier, but you can't just go to sleep at 6 p.m. out of nowhere.
Kenza, I'm interested what your answer to Vinod's question was.
For the coordination number.
Yeah.
Yeah, so the coordination number for the blue scale, so for the red scale, it's at 3.8 with a spread, obviously. And for the blue scale, it's at 3.5 with a bit of a wider spread. But I think the coordination number is really hard you can't really put much trust in it, because when you reconstruct the structure, sometimes you lose a strut here and there. So I like to look at the angles. I think they tell us more. And the perfect tetrahedral angle for connected struts, it's 109.5. And for the blue, we get an angle of 111. which is pretty close, with not that much of a spread. So I think it's safe to say that we're mostly looking at tetrahedral arrangement throughout the structure, because it would be 120 if we're looking at free connected nodes.
Nice, thanks. Wow, yeah, it's very close. It's closer than. Were you surprised by how close it was, or?
I was happy. It's something I could work with.
That's good.
But yeah, it took a couple of tries to get the tomography right and to get the analysis right. So we had much more of a spread at the beginning, but it was mostly because the reconstruction was not great. So the better we could get the reconstruction, the easier it was to get the numbers out of it. And then we started to get pretty nice distribution of values.
Right, doing that fib is really hard. That was amazing, the images you made. Really.
Yeah, the resin filling, I didn't waste too much time on it because I could see that I was not going anywhere with it because the imaging was really, really tricky. So the platinum filling was really a game changer.
You mentioned like the negatives of using X-ray tomography. What about micro CT?
It's just not an equipment we have in-house.
Oh,
when
okay.
I said drawbacks, of course, there's drawbacks to every technique. There's drawbacks to FIP too. It's destructive. You can melt the structure, and you also need to have a FIP. So it's just, I think more people have a FIP than people have a synchrotron. So that's a bit more accessible in this way. But of course, every technique has this drawback. And the resolution is limited in the FIP. So for us, it's fine. That's what we need. But you can't really go below 10 nanometer, which might be a problem for biological structures.
Kenza, I have a question about the FIB. So do you use like a specific setup to kind of mount the scale? I'm asking because at some point I wanted to kind of like use the FIB to alter some things in the kiting of the beetle, like kind of make holes or stuff.
Yeah.
Have you used that?
It's just a stub like you would use for SEM with then carbon tape on top, and then we just scratch some scales off. So you have more than a couple of scales on the stub. And then, yeah, it's exactly like an SEM basically. So we just put it on a platform and then you have those two beams. You can totally make holes or anything with a FIP. Holes, array, cuts, it's pretty versatile. You can also make very thin slices for the TM.
Nice.
How do you do the very thin slices of TEM? They just fall off onto like a grid.
So I didn't do it myself. We do have a TEM, but as I said, we just don't have a protocol for sample preparation.
Sure.
Because you need staining, and it's done with osmium usually, and we just don't want to use that here. And you need to put it in a resin block and then take the slice. And often it's a blind process because you don't really know where you're cutting. So here with the full reconstruction, you have an infinity of cuts, which is super powerful, instead of spending a whole day to make one single slice.
So true. I feel it.
So there will be 2 more minutes before we start. So please take the last 2 minutes to get some coffee or tea. And then next I will switch with Amanda. So then Amanda will be, introduce you guys.
All right, so almost time. The next speaker is Janis Heuer. Are you here? And you can go ahead and start.
Yes, I'm here.
Okay.
So I hope you can see the slides. So good morning, everyone, from 7 o'clock in the morning in Germany. My name is Janis Heuer. My topic today is correlated characterization of nanostructures for anti-reflection on the wing membrane of butterflies. And so I'm from the KIT in Karlsruhe, and this is a work in collaboration with the University of Buenos Aires in Argentina and the Natural Science Museum in Buenos Aires. And first, I would like to talk about what is actually correlated characterization. So here you see the surface of such a glasswing butterfly that I'm investigating in this work. And we now would like to maybe know what this particular region is, or would like to investigate this more with different kinds of instruments. So let's say, for example, I would like to know the reflection. For this, I would need to go with the optical spectroscopy to this area and measure the reflection spectrum. And then I might want to know the topography. For this, I could use the white light interferometry. And now I might want to know really nanoscale topography. Then I would maybe use the AFM, the atomic force microscopy. And then I might have some certain features on the surface where I might want to know the chemical composition. For this, I could, for example, use the TOF SIMS. And of course, I might have some other scientific questions at this particular area on the surface. And then there's, of course, many more techniques that could be used. And so the key idea of correlated characterization is linking different measuring techniques together at the exact same spot. So really measuring at this location on the sample. And so what are the challenges of correlated characterization? Is that when we go from one instrument to the next and we place the sample into the machine, into the instrument, we can always have some sort of rotation or tilt that occurs. And we see here with this blue square that this rotation or tilt shifts our region of interest, our area that we want to measure. And so as a little example, if our sample is only 1 centimeter long, then we can, with a 5-degree tilt, we actually have a shift of our region of interest, or ROI, of 76 micrometers. And so this doesn't sound much, but if you're measuring with the AFM, your area is only a couple micrometers big. That means you might have lost it completely, or it's completely shifted somewhere else and you can't find it anymore. And so the real challenge is achieving a correlation at the nano and micrometer scale between these instruments. And so what is the solution to this problem? Is how can you locate the region of interest, ROI, in every instrument that you desire? the solution is to actually have 3 reference markers located around this region of interest. And so why would you need 3? Because we are in 3-dimensional space. That means that these 3 reference markers are located in 3-dimensional space, and also our region of interest is located somewhere around this. And so the solution would be to use these 3 reference markers to set up a plane of reference, which gives us all the information about the tilt, the rotation, etc. And with this, we can easily locate our region of interest. That means that when we go to our next instrument, our instrument 2, we again measure our reference markers first and set up our plane of reference. And now we can easily locate the region of interest because we've referenced it before. And so I've developed different softwares that calculates this for you automatically. It uses vector algebra. And so it gives you the coordinates basically where you find your region of interest based on the reference markers. And so to use this technique now, I use the example of these glasswing butterflies. So I investigate the anti-reflection of these glasswing butterflies. Specifically here, I will show you the Greta Otto butterfly, as you see here. And so I will measure one area with the atomic force microscopy, which gives us this nanoscale resolution with these nanopillars structures on top. And on this exact area, I also measure the reflection. So how good is the anti-reflection at this area with the optical spectrometer. And then as like the main motivation of the study is then can a simulation predict the spectrum? So the idea is that this topography, this nanoscale topography creates this anti-reflection spectrum. And so we kind of input the AFM data, this topography data into our simulation, and it should calculate this spectrum that we also measure experimentally. And so another challenge to this study, specifically for the glasswing butterflies, is that we measure on both sides of the wing, on the top and the bottom. We measure the reflection, the transmission, and the topography. And so how does this now look actually in practice is that here we see our light microscope image of the glasswing butterfly surface. And we see here these black dots around. So these are actually the roots of the bristles. So the wing has some hair-like structures that are bristles. And these are the roots, these black dots that you see. And they serve as ideal reference markers because they're very unique, identifiable features on the surface. And so I use these as reference markers. And then here in the middle, we see our area with the optical spectroscopy, which is 10 micrometers big. And on the same area, I also measure with the AFM, which is 8 by 8 micrometers. And so the experimental results show that we see these nice little nanopillar structures on the top side and on the bottom side. So they're on the same area because these transparent wings, these reference markers are seen on both sides. So that's why we can measure the top side and the bottom side very precisely. And we see that they're actually quite similar structure-wise, what you would probably also expect. And we see these nanopillar structures, which have like a triangle-like shape of around 300 nanometers roughly. And we see here on the right our height scale and also this sort of histogram distribution, which is important in the next slide. This is the depth histogram of this AFM data that I just showed you. So it's basically the height distribution. It takes the whole topography map and puts each height value into a histogram. And so we see here basically your average distribution of these nanopillar structures, the height distribution, is pretty much the same for the top, the red curve, and the bottom, the blue curve. And so after I've now measured these topography data, I go to the optical spectroscopy and measure the reflection. And here we see the reflection spectrum that I measured there. We see that the top side somehow has a lower reflection varying between 2 to 4%, depending on the wavelength. And the bottom side has, for some reason, a higher reflection, varying about between 4 and 6%. And so now in the next and final step, we would like to do optical simulation on this area, which means we take this AFM histogram that I just showed you and put this into our simulation. And it calculates there an effective volume fraction, which basically means that it creates this curve that is says the volume fraction basically is at 1 when you're inside the wing or just on the edge of the wing. And as you move away, the volume fraction reduces in distance due to this nanopillar shape that gets smaller at the top. So your effective volume fraction reduces as you remove away from the distance of the wing. And so from this curve, it calculates a refractive index profile. And here I actually take the top and the bottom side measurements of the AFM to merge this into one refractive index profile. And from this profile, you can calculate your reflection spectrum. And here you see the dashed line is my simulation compared with the experimentally observed, the red line. And so it matches actually quite nicely, but there's of course still room for improvements to really shift these hills and valleys on top of each other. But the simulation is already quite in the ballpark of what we measure actually on this area. And so the outlook of the correlated characterization is the integration of many more instruments. I'm also working on the TOF-SIMS currently and the Raman spectroscopy. And I've done already SVM measurements quite nicely. And of course, another key motivation is to investigate the heterogeneity of the surface. As you see here, this is already preliminary results of the Cosmosoma auge butterfly that we really have in a small area going on different spots, a quite significant change of reflection here, as we see with these three spectrum. And of course, then improve the simulations in the future. There's always work to do. Yeah, so thank you for your attention. I'm happy to answer any questions.
All right, thank you very much. We have about 1 minute. Does anyone have any questions? I was wondering if when you were taking the reflectance measurement and the AFM, if it covers the same average area as far as size goes.
Yeah, I can go quickly back to this one picture here you see. So it's okay. This is also true scale, so it's really the 10 micrometers on the 8x8 on the same area. So
okay, right. And then I had another question. The refractive index profile. So is that basically an average of all of an average of all of them, all of the nano shapes?
Yeah, it takes the whole 8x8 micrometer map and takes all of this area into this refractive index profile. Yeah.
All right, that's neat. Thank you very much. Are there any more questions? Okay, so with that, we can go to our next speaker, who is Pascal Freyer. You can go ahead and share your screen.
Good morning, good evening, good afternoon. Can you hear me?
Yes.
Okay, I'll try to share my screen. So, can I start? Good morning, everyone. I'm glad to present. Oh, now I'm hearing myself. So let's try again. Good morning, everyone. I'm glad to present some of the work we've recently been doing at the University of Groningen about the structural colors in starling feathers. And here you see the common starling, also known as European starling, at the top, and Cape glossy starling at the bottom. And we have compared these in this study. So if you look more closely at the European starling, there's a range of colors between blue, green, and purple. And we have, I show you here some of the close-ups of the feather in the middle, and on the right-hand side of the barbules, where you can clearly see the single barbule cells, which can vary quite a lot in color. And Cape starling, you have a similar situation with a range between blue and green. So if you look at the anatomy of these barbules, so here at the top you see a cross-section of the barbule. On the left-hand side, for the European starling, we see that there is many solid melanosome rodlets, and they form a neat layer close to the surface. And there's also a keratin cortex between the surface of the barbule and the start of the melanosomes. And we have confirmed this in panel B with SEM microscopy. And in panel C and D, you also see well, in C, you see an optical oil immersion microscopy image, and you can clearly see the rodlets. And for the Cape starling, you can see a similar thing. So the top two, so the top TEM image has been taken from literature, which is from a different Lampertonus species, but it shows a bit more clearly how the anatomy looks. And panel B again is the confirmation that this anatomy is actually present. And again, C and D is an optical microscope and another SEM microscopic image. So just to highlight the approximate structure for the European starling, we have these rodlets and with a cortex layer above. And for the Cape starling, we have supposedly platelets, but we see also in the optical study that these platelets sometimes are a bit elongated and thin. So it might be also a combination of both platelets and rodlets. So in the past, there have been great anatomy studies by Dürer and Craig and Hartley, and also very excellent evolutionary studies done by Meyer and co-workers. And we are hoping to build on similar studies that have studied single layer of melanosomes in barbules, and to understand more the broader mechanistic structure versus spectrum relationship, especially if we go to more advanced structures. So I can summarize my research question is basically to understand more the nanostructure versus optical spectrum relationship. And when we add more complexity to this, and maybe a sub-question is also whether this effective medium multi-layer approximation that we are using is appropriate for this study. So quickly going into the modeling method. This is a bit much in one slide, but I'll try to be quick. So on the left-hand side, you see basically the structure. And on the very left is the way that we approximate this anatomy by a varying refractive index.
We lost your sound, but I see that you are muted. Um, let's see. Let's wait a bit and see if Pascal can get back on. So the next speaker is scheduled to talk at 10:24, or well, that's my time. And we should probably just wait and see if Pascal can come back on. But Viola, will you be ready at 10:24?
I'm in principle ready, but I saw that we have the 10 minutes break scheduled afterwards, so maybe we can use that and shift all the talks.
Yeah, great, we do. We have an extra break in this part of the session. So, but if people are coming from the other session, we'll try to keep on schedule. And then if Pascal comes back, perhaps we can have him talk at the end.
Yeah. Hi, everybody. I'm just speaking to Pascal on the phone, so he got disconnected via internet. So he says that you can just continue and go on with the program.
Okay. Oh, and I mean, if you can let him know that there is
He can hear you now.
Okay. There is a slot of time available that we are just going to have a coffee break at my time 22:48 California time. So if he can get his internet working and he wants to try to come back, then we'll have a break. Okay. But still we should wait a couple minutes for Viola, just in case other people from the session will join.
Okay. I'm gonna try if my screen share works in the meantime. Can you see the slides, I hope?
Yeah.
Yes. All right.
Oh, there was a question for Janis, if Janis is still here in the chat.
Maybe Janis already fall asleep.
Yeah, I see Janis there.
Can we ask the next speaker to check out whether he's here? Dimitrije?
Dimitrije, are you here?
Yeah, I'm here. Yeah, I'm here.
Perfect.
Great. Okay, so the next speaker will be Viola Bauernfeind. And you can go ahead and start.
Thank you very much. Hello, everyone. My name is Viola, and I'm very happy to now discuss coloration in Sternotomis amabilis longhorn beetles. Longhorn beetles are spread all over the world and comprise around 35,000 species. Many of them carry extremely long antennae, and most of them feed on wood and may even occur as pests. But the reason that we are interested in them is their diverse color displays. Their color is often due to colored scales that adorn their insect body, as you can see here. And the color in these scales is often due to a combination of pigmentary and structural color. In terms of photonic structures inside these scales, different types have been identified so far. One of them is this ball and stick arrangement of chitin spheres that you can see here on the right-hand side, and another type are triply periodic bicontinuous surfaces, which you can see in picture I here. In longhorn beetles, these bicontinuous surfaces have only been found in the Sternotomi tribe so far. And recently, my group reported another type of photonic structure inside scales, which are sheet-like arrangements of air and chitin layers. The longhorn beetle I'm studying is called Sternotomis amabilis sylvia, and it's displayed here on the left-hand side. The body size of the insect is around 25 millimeters, and as you can see, this insect displays a slightly iridescent bright green base color with black stripes running horizontally and orange patches on the wings. Under the light microscope, we can see that the black elytra is covered with colored scales in green as well as orange regions. If we take a closer look at this green-blue scales over here, we can see that the scale seems to be very shiny and bright green at some part, which is the base where it's fixed on the elytra, and the rest of the scale seems to be a very dull green color. Similarly, in the orange scales, we see that the base is relatively bright, but does not have any shiny appearance, and towards the end of the scale, which I will call the fin, it displays a very dark orange. To assess this difference, I performed microspectrophotometry reflectance measurements, where we can see that the peak wavelength position is more or less independent of the position that we measured on the scale, either on the base or on the fin. But in the case of the green-blue scales, we see that the intensity slightly differs. I also performed transmission spectroscopy on these scales, which confirmed that the coloration is due to a combination of pigmentary and structural color. Scanning electron microscopy gives us further insight into the scale geometry. On top, we can see SEM images of green-blue scales from top view and side view, and below we can see orange scales, which are on average slightly larger than the green-blue ones. However, they both share the same geometry with a relatively flat base, and towards the end, we can see here very nicely in side view that they have a triangular extension of the cortex, which is vertically away from the elytra, which I will call the fin, and this fin is further decorated with those more or less parallel running ribs in both types. Now comparing the side view images with high magnification light microscopy images, we find that the color originates only from a certain part of the scale, which is not covered with ribs. And this is what I will call the scale body. And so to understand where the coloration of this scale body is coming from, we have to look inside the scale body. Scales that broke open during sample preparations allow us to do so. And this is the green-blue scale. We can see that the inside is filled with an ordered periodic network of sticks that are connected. And this network is wrapped in the cortex, which folds into ribs and microribs. Now we want to figure out what kind of network we're looking at. And for that, we take a closer look at this element over here, where we can see 7 sticks that connect individual elements to another and to the plane behind. However, there is no 7 connected network. So for symmetry reasons, we can impose that there is another stick behind this element over here, which we cannot see from this perspective. And thus, we assume we're dealing with a 8-fold connectivity. What is very dominant in this SEM image here is this cross pattern, which I indicated in blue, and the sticks that seem to point outward of the plane towards us, which are indicated in yellow. And with these considerations, I figured this could be well described by body-centered cubic symmetry, just sketched here, and based on which I designed a model of a BCC crystal, and tilting this model to the 011 direction, I could actually successfully reproduce the pattern that I saw in the left-hand side SEM image. Also, the 001 direction has been observed in this network, as you can nicely see here with the comparison of the left-hand side illustration and the SEM image. If we were to describe this network now as a triplet periodic bicontinuous surface, it would correspond to the IWP Schön surface, which also shows eightfold connectivity. Now the situation is slightly different for orange scales. And as you can see here in this SEM image, the inside is now filled with a network of sticks of more or less similar diameter and length, but they seem to be connected in a very random way. So I would call this an amorphous quasi-ordered network. To gain further insight into these networks now, which I'll call photonic networks, I performed focus ion beam milling to obtain cross sections of these scales. And these now nicely show that these photonic networks are indeed restricted only to the scale body and do not extend into the ribs. At higher magnification, we can see for the green-blue scales that they are indeed filled with photonic crystal, which shows several domains. And therefore, I call this a polycrystalline photonic crystal with body-centered cubic symmetry. In contrast, within the orange scales, we cannot discern any domains whatsoever. Therefore, we term this a quasi-ordered amorphous network. This gives us now the base to understand the coloration of the scale body, but we do not understand the function of the fin so far with those ribs. And to do so, I performed case-based imaging to study the scattering behavior of an individual scale. Here we can see that for low angle illumination and high angle illumination, corresponding to 15 and about 64 degrees, we find in the green-blue scales that we have for low angle illumination several spectral reflections, which is what we expected based on the polycrystalline nature of the photonic crystal inside the scale. For high angle illumination, we obtain a very gradual shift from green to blue for high scattering angles, which is characteristic for iridescence. Repeating the same measurement at the fin, we find that the signal is distributed to a larger range of angles, and for high illumination, we obtain that the iridescence is lost to a very large extent. Therefore, I assume that the fin presumably acts as a diffuser. Now, the situation is different for the orange scale, since we know beforehand that these are filled with an amorphous network. Therefore, we do not expect any spectral reflections. And this is also what we see here. We have a more or less random distribution of intensities over here. And for high angle illumination, we obtain a very homogeneous beige, bright orange with no signs of iridescence. Here, the presumably diffusing character of the fin does not really change the scattering characteristics. To confirm now the hypothesis of the fin acting as a diffuser, I performed FDTT simulations on a 3D model that I shaped according to this scale fin, where the ribs are inclined at a certain angle alpha. And in these 2D simulations, the material refractive index was set to the one of chitin. I used perfectly matched layers as region boundaries, and a Gaussian beam served as illumination from below the structure to mimic the effect of the photonic crystal, which is in fact below this fin structure. From these simulations, I calculated the far field projection in terms of scattering intensity over angle for different wavelengths. This is now only the source for reference. High intensities are in red, low ones in blue. If we now add a triangularly shaped fin above this Gaussian source, we obtain this result, which indicates enhancement in scattering. And if we now decorate this triangular fin in addition with those ribs, we observe that this effect is even increased. With that, I would like to sum up the results that I just presented. And we find that in the Longhorn beetle, Sternotomis amabilis sylvia, coloration is due to scales which are colored themselves and adorn the insect's elytra. The coloration of the individual scales is produced by a combination of pigmentary and structural color. And this structural color is due to photonic crystals inside the scale body, where the green-blue scales are filled with polycrystalline photonic crystals of body-centered cubic symmetry, and order scales are filled with quasi-ordered amorphous networks. Finally, we also studied the influence of the scale geometry and found that the scale fin acts as a diffuser and thus suppresses iridescence. With that, I'm at the end of my talk and would like to thank my supervisors and colleagues for helpful discussions, our funding agencies, and the organizers of this conference for giving me the opportunity to talk today. And of course, all of you, thank you very much for your attention.
All right, thank you very much, Viola. Are there any questions? Let's see, our next talk begins in about 2 minutes. So.
Hi, Viola. Hi, hello.
Hi.
Yeah, can you just explain me once more about that numerical simulation which you did for that just
sure.
I didn't got, yeah.
Sure. So I'll just go back to that slide for this one or the previous one. So I used a Gaussian beam because obviously we know that illumination from natural light is coming from above, but we were interested in studying only the effect that the ribs would have from illumination below, because we know the photonic crystals select certain wavelengths. So we wanted to disentangle those two effects. But obviously, if we have illumination below, this would basically increase the angles of illuminating light reaching the photonic crystal in the end. So we would have this effect, the scattering effect, in both ways, obviously, and would lead to more homogeneous color in the end. Is that clear?
Increasing that thing, well, the third simulation, what it signifies in comparison to first one? What is the difference? I didn't got. Like, what is the difference between the third simulation, which you did for source plus triangular field plus ribs, in comparison to normal? Yeah.
So we can see this is the source only for reference. And then if we add this triangular shape above the source, we see that scattering is increased and scattering occurs at larger angles. And then in addition, putting these ribs on top of this, we get scattering of way above 50 degrees. So we can say that intensity is scattered to larger angles, and therefore we obtain a more diffuse signal.
Thanks.
Sorry for not making this clear.
There's a couple more questions in the chat, Viola, if you want to answer them, but we'll go ahead and move on to the next speaker.
Right, thank you very much.
Thank you. And so the next speaker is Dimitrije Mara.
I hope you see the slides. Okay, so I can start them. Good morning to everybody. Today I will talk about linear and nonlinear optical response from the Cisada transparent wings. Okay, so first I will talk a little bit about fluorescence and second harmonic generation to explain a little bit. Fluorescence comes from the different molecules who have ability to absorb and emit light. In this case, in the insects, we have most probably some organic molecules which can absorb the light. We can have two different ways how we can excite them. One is linear or one photon excitation, where we use high energy photon from the UV or visible part of the light, which excites singlet electrons from singlet ground state to singlet excited state. And then these electrons can be radiatively relaxed by emitting the light back to the ground state. The second way how we can use excite also is with two photon excitation, where we have two high power, lower energy photons in the, let's say, near infrared, around 1000 nanometers, where we have so it absorbs first one photon to a virtual state and then second photon to the excited state. And from there, it has the same possibility to relax, radiatively emitting one photon back from it. And on the other hand, second harmonic generation, there we have some rules that we need to have to possess material to possess second harmonic should first absorb two high power photons similar to the two photon excitation. Just in this case, simultaneously it will emit the third photon, which is higher energy, and so if we say half of the wavelength that we use, in the case if we have, example, 1000 nanometer photons, the emitted photon would be around 500 nanometers. And also, to have second harmonic generation, we should have non-center symmetry in our samples. Most of the fluorophores that we can find in beetle wings and something are like butane, diclofulin, papillon, green fluorescent protein. What is for all of these chromophores same? It's that they have conjugated double bonds. So these double bonds are what we can, what absorbs the light and emit. Here are presented the beetle wings of Cicada orini and Plabeus gray, which we used, beehawk moth, and Dempsey fly. First, I will talk about some structural of this study. So on the left, we can see the optical microscopy under the visible light and under UV light. We can see that in both we have the emission of both in the transparent part as well in the veins of the wings we see. Of course, we see a much stronger emission in the veins due to most probably higher concentration of heating and resin inside. On the right, we can see Cicada orini, the bee moth, and Dempsey fly wings under SEM on A, C, and E top, and B, D, and F on the side. It's interesting that Cicada and bee moth have, let's say, conical cylindric shapes with different distances inside, while the Dempsey fly has amorphous, we can say amorphous like construction of the field. Then we did it linear and linear spectroscopy fluorescence, where we excited a different wavelengths from 320 to 440 nanometers, where we can see the shift of the emission also with the excitation in all examples, where we have somewhere stronger emission and harder shift, more to the right, depending on the type of Cicada orini, Plabeus, or moth, or Dempsey fly. We can see that, for example, Dempsey fly, we have significant decrease and stay at the same wavelength, and then moving when we excited at lower wavelengths, and then increase and moving the array. Also, we measured the lifetime measurements of all of this, and here is the example of bee moth wing excited at 340 with emission at around 400 nanometers. In all cases, we had pi exponential fitting with shorter lifetimes around 1 to 2 nanoseconds and longer lifetimes between 7, 8 nanoseconds range. What is interesting, we also saw some second harmonic on the right and multiphoton emission of Cicada orini and L. Plabeus transparent wings. We can see that in both cases, the veins show much stronger signal intensity in both second harmonic and multiphoton, while in multiphoton we have less intensity from the transparent part, while we see much stronger second harmonic response. This can be due to the conical shape, which is really disordered and non-center symmetric. Here we can see how the Cicada orini looks under visible light, under white light, and also under UV light. We can see really how nicely it also reflects and emits. And for conclusion, what I can say is that we showed that all wings have shown emission under the UV light excitation and visible light. Also, we compare this with the Dempsey fly as our control, because that's already known results. And the wings of Cicada showed second harmonic and 2 photon response, which with veins which have most probably higher concentration of resin hidden inside. And we showed that nonlinear optical characteristics, both of these other wings and other, can be used as an additional technique regarding information of fluorophores and characterization of the insect materials in general. I would like to thank you all for attention, and also I would like to thank the KU Leuven for funding my postdoctoral fellowship, as well as co-authors from this. And preprint will be soon on archives. Thank you.
All right, thank you very much, Dimitrije. Are there any questions? We have about 3 minutes. Dimitrije, do you know if there is any biological function to the fluorescence of the species that you studied?
So it's as a biological material. I'm a chemist, so from the chemistry side, I can say we cannot know what kind of fluorophores we have inside. So if we want to, when we excite in UV, we can also have in that part also, if there is some proteins can also, if we have any kind of tryptophan in proteins, we can also see that emission, which comes from around 320, the excitation is, and they emit around 380. So we should extract, but unfortunately, due to small concentration of materials, we cannot say what kind of fluorophores, because that's a combination of different varieties of fluorophores, and the lifetimes of them are more or less always the same. So it's really difficult to know if we have one or maybe 2 or 3 different fluorophores inside the wings.
Okay, thanks. Looks like we have a question. So Rox, is this to Dimitrije?
Yeah.
Okay. Are you able to extrapolate from the variation in the molecules that you can see using UV to understand the physical characteristics of the insect wings? And what sort of physical characteristics might you be able to measure?
So to extrapolate it would be difficult. We should have the pure molecules, let's say dyes that we suspect that we have inside, and measure them, and then maybe do some extrapolation of under the curves to see if we can see what is what. But that could be quite difficult, because it can be a mixture of known and unknown fluorophores, which would be really difficult to understand if we don't do, let's say, an extraction and try to really extract from the wings the molecules, and then use the pure ones and know what we have in the physical characterization, because that's what is possible without damaging the wings, of course.
Okay, great. Thank you very much.
You're welcome.
I believe I saw Pascal here. But
yeah, I talked with Pascal. He said he will be ready, yeah, after you call him. I don't know whether his internet is okay now.
Okay. So we can use this time unless Pascal.
Oh yeah, he replied to me. He said he's here. Could you unmute yourself and share your screen, Pascal?
Yes, can you hear me?
Yes.
Excellent. So I'm sharing my screen. Okay, I hope you can see this. So this is where I left off. I'm sorry for the little intermezzo. I had some hardware problems. So I left off with
So, sorry to interrupt. Since we have enough time, maybe you can start over. You can go faster for the previous part you talked.
Certainly,
Sorry about
certainly.
this.
No problem, no problem. I think that's better for the recording. So I would like to share some of the research we've been doing on starling structural coloration. And we see here the European starling at the top, also common starling, and known as common starling, and the Cape glossy starling at the bottom. And if we look more closely at the European starling feathers, we see a range of colors between green and purple. As you see here on the right-hand side, we have close-ups of the feathers and also of the feather barbules, where you can clearly see the cells, barbule cells, which sometimes have very varying coloration. And for the Cape starling, we have a similar story. The color varies not as much as for the European starling. So we have a gradient between green and blue, most predominantly blue though. So looking at the anatomy, a cross-section of these barbules shows solid melanin rodlets that are ordered very well spaced from the surface of the barbule here on the left-hand side. And the top figure has been from literature, which is not, it's only a related bird with the same structure, taken from Dürer. And panel B, C, and D, we have acquired to confirm this structure in the European starling. On the right-hand side, similarly, the top figure is a related bird, also taken from the same publication of Dürer. And at the bottom, we confirm this structure in the Cape glossy starling. What is nice to see also in panels C is the optical microscope image with oil immersion, and you actually see the distribution of melanosomes very well there. And we see that it's not only platelets, but also elongated platelets. So we cannot tell whether it's actually just a disordered arrangement of same-sized platelets, but it seems like from the SEM that we have taken in panel B, that it's more a distribution of rodlets between rodlets and platelets. So there's a mixture or a transition going on there, I would say. So there has been a lot of anatomy study and also evolutionary pathways, really nice works, and also very nice works on very similar structures, also a single layer of melanosomes that is spaced from the cortex. And they have already concluded that the cortex plays an important role. But I hope to build on this with a more broader look at what happens if you add complexity with the hollow melanosomes, for example. So we have modeled these structures, and on the left-hand side, you can see basically a schematic which shows the solid melanosomes as black, and the cortex parameter and the diameter. And in this figure, basically the surface of the barbule is the top, and the light comes in and is reflected from these interfaces. And we model this by an effective medium. So we basically average the refractive index, and we make a uniform refractive index stack, as you see here in the middle. And from there, we can compute the reflected and transmitted components of light using a transfer matrix method, which is very nicely described in the of Ye in 2005. So here we come to the results of the European starling. At first, at the top, you see measurements of microspectrophotometry, more or less of the regions that are shown, so green and purple. And you see, of course, some variation. One of these spectra will certainly look more bluish than green. But we have looked here at the main colors that the bird possesses, and have modeled these. So at the bottom left, you can see for 2 selected spectra, we have basically fitted these with modeling to determine the possible parameters that explain this spectrum. For the Cape starling, we did the similar thing. We also see a variation of coloration, which you can see also in the image above, where probably the right cell, which looks more purplish, is actually one of the spectra where the blue peak has gone so far into the short wavelengths that the red becomes more present. Here again, we have modeled, the schematic is in the bottom right, of the anatomy with the cortex C and a thickness to the melanosome membrane and the air gap in between. And we can find nicely corresponding spectra shown here on the left-hand side. So to conclude, we were able to model these spectra. Here again, I show the schematics. The light basically comes from the left-hand side, it passes the cortex and the layers. And we conclude that for the European starling, so the simpler structure where you have only the cortex and the melanosomes, we saw that the melanosome thickness is not relevant to the spectra that we measure. We do see in the modeling, which is a very perfect ideal situation, that we see, of course, spectral variations, but these are not to be seen in the measurements. So we conclude that really the cortex is the only parameter relevant for these color variations between purple and green. And this also confirms previous studies of similar structures. If we go to a more complex, basically 4-layered nanostructure with the cortex, a membrane, air layer, and another melanin membrane, we see, as we expect from multi-layer theory, that also the other layers are more influence the spectrum more strongly. But we can conclude also from anatomy, where it is shown that the variation of the melanosome shape is not very large, and also from the fact that the cortex influences the spectrum far more than a variation of the melanin and the air layers. We suggest that also there the cortex is actually responsible for the color variation. Yes, I'm always very, very positively surprised how well this modeling, multi-layer modeling, can describe the bird spectra. And I hope that this inspires also more people to look at this in a more broader comparison, as this humble initial start between 2 species has shown. So this is just the text version of my conclusion. And with that, I would very much like to thank my supervisor and co-workers, Dukle and Bodo, and also Hein, for technical support. And last but not least, thank you so much, Living Light community, and to all of you listening.
All right, thank you very much, Pascal. We're glad that you got to get back up here. Are there any questions? We have about 2 minutes. Okay, question from Rox. Thanks, Pascal. Why do we see the edge outlines of the hollow melanosomes in optical microscopy rather than reflection from the whole melanosome surface?
That's a very nice question. I'm not sure to give the short answer. Well, at the outlines, you can certainly see that there's more melanin there, so that might certainly influence that view. But yeah, I cannot give an explanation for why that is the case.
Great, thank you. Are there any more questions? Do you have a method of showing like the goodness of fit of the reflectance spectra with the model?
That's a nice question, and that's also what all reviewers often ask me: is there not a more objective way instead of looking by eye which parameters fit most? It would of course be nice, and also if you want to include things like pattern recognition and machine learning to analyze a lot of data and compare that. If you have some computational way of getting the best fit, and I'm sure there is. The only problem is that between measured spectra, the intensity of these spectra varies a lot. So there's a lot of uncertainty there, and I'm not sure whether a fit would be adversely affected by this intensity uncertainty, because in the end, if you fit a spectrum, it will also take that into account, I believe. And furthermore, we learn a lot from actually looking at what happens if we take one parameter and we vary it. And that is really also, I emphasize, a very nice part of this modeling method. Actually, I was playing with the thought of inspiring you with some kind of a sequence of parameter variations. But I think that one would have to take another dimension of presentation there, which is more a movie kind of presentation, what actually happens, because it's a very dynamical process and also complex. And what you say is right. How can you take away the factor that you get lost into some details?
All right, great. Thank you very much. With that, I'll pass it back to Ming. I believe we're going into flash presentations now.
Yes, I will start sharing the presentation. Pascal, could you stop your sharing?
Oh, yes.
So before we start, let me make sure all the three presenters for Flash presentation are here. So Junwei,
yes, they are and they are ready.
Okay. Oh, they are all ready. Perfect. So yeah.
Russian is around. He didn't reply to me, but we can go.
Okay, so this is a Flash presentation. Please try to finish the presentation within 2 minutes. I will have a clock here, a timer here. I will stop you at 2 minutes sharp. So at the end of all the three presenters, we will probably have a few minutes to answer a question. Yeah, so yeah, Junwei, are you ready?
Yeah, I am.
Okay, you can start.
Okay, well, let me start my timer as well. Thank you for having me here. I will talk about structural color of colloidal clusters. So we know nature produces fascinating colors, and they not only produce it in a large scale, and they also produce it tiny bits of opals, and this is what we're trying to do. The way to do it is to simply put a drop of colloidal dispersions in oil and then let the colloidal dispersion dry, as you can see from the screen. As the water droplet dries, the colloidal particles come closer, and then at a certain packing fraction, they will start to crystallize and form a crystalline colloidal cluster that is discrete and that's crystalline and that is dispersible in water. Well, in oil, dispersible in liquid. You can transfer them back to water later. So when we examine closely what the cluster look like, we will see that there's 3 types of clusters. What I showed here are 2 major types. One is the icosahedral cluster, which you can see from the on the upper right corner, and also the lower left. And another type is the FCC cluster. The icosahedral cluster means that they have 20 domains of face center qubit lattice arrangement. So these 20 domains, these are 20 tetrahedral domains, and then each domain looks like what is shown here as a green tetrahedron. And then when you examine this cluster from different orientations with different incident light angles, you will see different color patterns of the cluster. As you can see from the left, lower left corner, you can see either a triangle if you look from the top at this 3-fold axis, or you will look at a bow tie structure if you turn the cluster into a 2-fold axis. And that's it. Yeah.
Okay, thanks. Let's move to next one is Charlotte. I don't see, oh, here is the name.
Yeah, I'm there.
Perfect.
Okay. I can start?
Yeah.
Okay, so hello everyone, I'm Charlotte Bouquier from Namur, Belgium, and today I will talk about the effect of the cholesterol content on the second order nonlinear optical responses of diethanaps embedded in phosphatidylcholine lipid bilayers. So first, we study cell membranes because of their importance. Lipid membranes perform diverse and essential functions in our cells, and those functions are controlled and enabled by the enormous lipid structure diversity. Here we focus on 2 main classes, glycerophospholipids and sterols, and more specifically, phosphatidylcholine and cholesterol respectively. The technique we use to study the membrane is the second harmonic generation, and it is a process where pairs of incident photons of frequency omega are converted into photons of frequency 2 omega, and this technique is specific to interfacial regions like the lipid bilayers. Most biomolecules possess few groups with exploitable nonlinear optical properties, so that we use exogenous dyes like the diethanaps to improve the contrast in the tissues. The first step in our work is to perform the molecular dynamic simulations of the dyed lipid bilayers and to extract snapshots, and then on those to compute the molecular SAT response at the QM level. For this, several systems were created from the pure DPPC bilayer to bilayers containing up to 66 cholesterol mole fraction. And by analyzing the structural parameters of the bilayer, we observed what is known as the condensing effect of cholesterol that aligned the lipid as well as of the probe, that induce an increase of the NLO response, and that will lead us to be able to have a tool to determine the composition of a given lipid bilayer based on the NLO response of the embedded dye. Thank you very much, and I can take questions in the chat.
Okay. Yeah, I'm ready if you are ready.
Oh yeah, you can start sharing. Yeah. Maybe somebody joined later, so this will be 10 minutes talk, and then we may have like a 2 minutes question. Yeah, if there are, and I will like give you one warning before like 1 minute. So we want to keep everything on track. So I think, yeah, the floor is yours. Luisa, go ahead.
Okay, I hope you now see my slides. Nice. Yeah, thank you for the kind introduction. My name is Luisa Borgmann, and I'm currently a PhD student at the Research Group for Biomimetic Surfaces and Scanning Probe Technologies at the Institute of Microstructure Technologies at KIT in Karlsruhe. And today I want to tell you a bit about the method I use to turn initially transparent polymers into bright white and scattering ones, which is thought to be maybe once some kind of replacement for titanium dioxide. And yeah, titanium dioxide is the most used white pigment for consumer goods of all kinds. So you will find it in paints, in plastics, but also in cosmetics and food. However, there is an ongoing discussion in recent years about possible health risks, if it's maybe carcinogenic to humans. And it has also been banned in France since 2020. Besides this, there is, as you can imagine, quite an energy-intensive extraction process when you go from a titanium ore to final pigment. And in the production, there are also harmful byproducts, which have now to be disposed environmental friendly, and this is also raising the cost of the pigment. Nonetheless, the pigment is not recycled so far. And it is used so broadly because it has a very high refractive index. So when you embed it in a matrix, yeah, the contrast in the refractive index will cause very high efficient scattering. However, in nature, we know from various examples that it's also possible to get very highly scattering surfaces and structures by just using a biopolymer and air voids. The most famous one has been discussed a lot, the white beetle, which I show here in the right corner of the picture, which has very thin scales that nonetheless can scatter very efficiently. And we took this as a benchmark and an idea how we could produce such air void polymer structures. And what we use is a foaming process called supercritical CO2 foaming. It has the advantage that CO2 is non-toxic, and compared to chemical foaming processes, there will be no traces of the solvents in the polymer left behind. And basically what I have there is an autoclave, which I can heat up to a certain temperature, and the CO2 is then pressurized up to 500 megapascal, which is 500 bar with a syringe pump. And what is happening to my samples in the autoclave is first that the CO2 is taken up in the polymer at a constant pressure and temperature, and thereby it swells. And it also starts to soften, so the glass transition temperature starts to decrease. And then I introduce a very fast pressure release. And under the new conditions, now the CO2 is no more soluble in the polymer. And as a consequence, the CO2 starts to form nuclei, and then more CO2 can diffuse into the nuclei and start to form pores. Part of the CO2 is also lost over the open surface. And while the CO2 is diffusing into pores or out of the surface, the glass transition temperature rises again. The polymer hardens, and when the polymer hardens, in the end it's stabilized. What we see here is now a foil, which has initially been transparent on the left side of the picture, of the bottom picture. And after foaming, it is opaque and white on the right side of the bottom picture. However, when we are foaming foils, we are very restricted to the dimensions of the autoclave. It's a batch process, so it's rather small. And we've been asked about options to integrate this more in existing processes. And there the idea was, instead of foaming foils, rather foaming particles. And I did so with PMMA particles here in a diameter of 60 micron. To have a look into the core of the particles, I put them into glycerine so that the reflections on the surface are suppressed and we can just see the inner structures. And as we can see, there is a temperature range where we get a very bright reflecting core. And comparing yeah, these particles formed at different temperatures. I found that the parameter of 110 degrees seems to be giving the most reflecting core structure. And we also had a look into the inner structure of this core. There we got some help from Christine Santos de Oliveira from the Martin Luther University in Halle-Wittenberg. And she analyzed the inner structure, and we can see here now one of those polymeric particles with this formed core. And with rising temperature, the pore dimensions also increase. And also the pore density, the volume fraction of the pores increases until it levels out at 12% approximately. And what we see here is that the pores between 200 and 300 nanometer are really in the range where we expect effective scattering, and also from literature, pores in the range between 200 and 250 nanometer have been suggested to be very effectively scattering when considering spherical air pores in some kind of resin with a refractive index of 1.5. However, as you can see, there is still this unformed core, unformed layer, which I wanted to reduce further. And there is a method to, instead of foaming the particles just purely in the autoclave, as I did, and which is shown in the upper panel, one can also put them in some kind of barrier film. I did so with a PVA solution, which is then dried and forming a film where the microparticles are embedded. And while when foaming the particles pure, they will lose a lot of CO2 at the surface. When foamed purely, they will lose less when they are embedded in this film, and thereby the unfoamed layer will be reduced. And afterwards, then one has to dissolve the matrix again, which is quite easy because it's possible in water, and one has to filter and dry the particles. And here again, I show the particles formed at different temperatures, again in glycerine to suppress the surface reflections. And what you can see here is that indeed the surface layer, the unformed surface layer has reduced a lot. And also the most reflecting samples are now achieved also at lower temperatures. One additional advantage of this method is that now it's possible also to form smaller particles of 40 or 20 micrometer, which have not been very successful in forming so far when not being embedded in a barrier film. So, and with this, I'd like to summarize. So I showed you today the method I'm working with, the supercritical CO2 forming of, in this case, PMMA particles. We achieved pores in the range of 200 to 300 nanometer, and we also reduced CO2 losses at the surface, so reduced the unformed layer by introducing a diffusion barrier. Next steps that we consider is to use biodegradable polymers and to further look into the barrier film properties by changing the material or add some plasticizing additives. And what you see here is a first try I made with the particles I foamed. I put them in some kind of transparently drying paint, so some kind of acrylic binder. Then put the particles in and really painted those beetles, once with the unfoamed, once with the PVA foamed, and one with the purely foamed particles. Yeah, and last but not least, I'd like to thank my research group for their support, and also all the institutes that have been included in this work, and also the funding, and of course all you organizers from the Living Lights. It was a pleasure. And then I'm open for questions.
Yeah, thank you for the nice talk. We have 2 minutes for questions. So here is a question. So from Surdar, I have one question. Did you consider the absorption in your data analysis?
And no, not that much, because what I used was PMMA, which is rather not known for being very absorbing in the visible part of light. However, it would be interesting to consider more the UV absorption, because there it might be that there is some absorption due to the manufacturing of those particles. Yeah.
Here's another question. Do you think the forming process be adapted to make a formed material that can be re-infiltrated by second phase?
I think I didn't completely understand the question.
Okay, it's in the chat. So
yeah.
do you want to read it?
Okay, that the pores are filled then afterwards with another material. I'm not sure, because the pores are rather tending to be closed, so that it's I'm not sure if there is something that can penetrate the pores.
Okay. I have one question. What is the mean transport length for your system? Pick up one wavelength, any wavelength, maybe at a 400 or 500 nanometer.
I actually don't know, sorry.
Okay. No. Okay, now it's the time to move to next. How
about now?
Yes.
Oh,
perfect. Okay, perfect.
Cool.
Good, right. Let me just turn these off and see if that helps. Okay, how's that? Great, sorry again. Yeah, so I'm going to talk about my work on using astrophysics to improve how we detect breast cancer. So I'm going to start with a little bit of the motivation behind that, which is the fact that in 2015 there were over 55,000 new cases of breast cancer in the UK and over 11,500 deaths. It's estimated that 1 in 8 women in their lifetime will get breast cancer. And so any way that we can improve the detection of that would be beneficial. At the moment, the gold standard way of detecting breast cancer in the UK is to go for a mammogram, which is where the breast is compressed down and then irradiated with X-rays in order to have a look at what's going on inside. And then usually a needle biopsy is performed, where they insert a needle into the suspicious area, take a sample out, and then send that away to be tested. Currently, about 80% of these biopsies come back as not being cancerous, so it's quite stressful for patients to go through the procedure and is quite expensive for the NHS. So we're trying to find a non-invasive way of reducing the number of needle biopsies that need to be performed. And where my work comes in is doing Monte Carlo modeling. So I'm going to do a little brief bit about what Monte Carlo modeling is and how it ties in here. The code that I use that runs on this type of modeling was originally used for modeling how light travels in the dusty disks around baby stars, which is how the astrophysics comes in, and that's my background. But now we're applying it to how light travels through the human body. So Monte Carlo modeling involves taking a photon and inserting it into a material and then moving it and following it through that material. So the first thing that you need to know is how big a step that photon is going to take through it. If you have something that's not very scattering, like a glass of water, it's going to take big steps through that. And if you think of something that's really scattering, like a glass of milk, it's going to take much smaller steps. So once we know our step size and we randomly generate a direction, we then move our photon, and then we can do one of two things. We just move it again and we keep doing that until it has escaped the domain, or it will be absorbed by the material. So you'll add the amount of energy that photon had to the location, and then we go back to the start and we release another photon, and we do that millions and millions of times and build up a statistical picture of where light is expected to go in this material. And this is a very simplistic view. Like, in reality, when we run these simulations, you can add in like lots of different optical properties and shapes and refractive indices and all the rest of it to get like a good idea of what's going to happen when you shine this light into this material. So in the case of my project, we're applying those photons into breast tissue in the human body instead of space, which we were doing originally. Which is all very well, shining light into the body, but we need to know what it is that we're looking for and what we're trying to find. And so for the case of breast cancer, we're looking for microcalcifications, which are a good biomarker. Microcalcifications are small calcium deposits that can occur in the breast for a variety of reasons, some malignant, some benign. They're too small to feel, they're not painful, but they can be seen sometimes on mammograms. So this one on the left here is like an eggshell calcification that would form around like an oily cyst or something, and that would be benign. And on the right, this is what we would call casting distribution. So you can see it's kind of like following like along a line, and this would be malignant. This is indicative of cancer. And in this bottom sketch, it's showing you how the different distributions can indicate the type of lesion that you've got present. So going from left to right, we're going from least cancerous to most cancerous. But it's not just the distribution that can tell us about what's happening in the breast, it's also the type of calcification that's present. So there are two types of microcalcification, chemically speaking, that we find in breast tissue. Type 1, calcium oxalate, or type 2, calcium hydroxyapatite. And calcium oxalate is not associated with cancer, but calcium hydroxyapatite can be, or so it can appear in both benign and malignant cases. So if we had a way of identifying which type of calcification was in the breast tissue, then we would be able to say that those with type 1 calcifications don't need to go for biopsies, and that would be a good way of reducing that expense. So we need a way of chemically identifying what molecules are in the breast tissue. And that is where Raman spectroscopy comes into it. So in like 99.99999% of cases, we have elastic scattering when light interacts with a molecule. So it comes in, it excites the little molecule, and then it goes away again at the same wavelength, same energy as it did in the first place. But 1 in a million times, we get what we call a Raman scattering, where that light comes in and excites the molecule, and then it goes away with a different color, so a different energy, because that molecule has either kept or given some energy to the photon. And the amount of energy that it keeps or gives depends on the type of molecule it is, so the size of the atom and the strength of the bonds. Big atoms and strong bonds will keep more energy than light atoms and weak bonds. And so by examining this difference in energy, you can tell what kind of molecule it is that you have in your material. So in a slightly more scientific way, we're changing the vibrational energy of the molecule. So here's an example of, we've got a pork chop with some calcification on it. So this is the Raman signal from the pink proteiny bit of the pork chop. This is the signal from the white fatty bit. And this is the signal from the calcification. And looking at these numbers, it should be very easy to tell if there is that calcification present in tissue, but it's actually not, because from this picture we've put absolutely loads of calcification on this pork chop, whereas in reality you're going to have like on the order of micrograms in the breast tissue. So we're trying to find this spike in amongst this mess, but when it's orders of magnitude smaller. So we need to look for a way of improving the probes that we have, looking at like the input beam size and where we have the detector and what size of detector that we have. So my current research work has been running models of different systems to try and find how we can maximize the amount of Raman light that we get out. So the setup that I've done in my simulation is having a block with the optical properties of pork tissue, an input laser beam, and then a little distribution of microcalcification. So I've gone for a 1 centimeter sphere that's got 10 calcifications in it. I move it around inside my pork chop block, and then I have an array of detectors to look at what would be the best one. So the first result here is if I have the laser coming in here on the left, and then a 1 centimeter diameter detector on the right, and then I move this through. So every one of these squares is having this distribution in a different position. And this X location is the optical axis. And it's for a 1 centimeter beam and a 1 centimeter detection. And so we call this transmission Raman because it's taking the bulk properties of the block. And we can see that once you move this distribution off the optical axis, the signal starts to drop off quite rapidly. And we're getting a maximum of about 10 to the minus 8 arbitrary units for the Raman detection. So remember that number. So on the next slide, I've shown what happens if we put a laser beam in and then collect in an annulus. So in this way, we're detecting the backscattering. And you can see it's a different shape, and we're not probing all the way through, but we are getting 2 orders of magnitude more signal in that area near the top. So it is also useful. So this is spatially offset Raman, sorry. And you can see that if we want to look at the bulk properties all the way through the material, then we want to use the transmission thing. And if we want to probe in more detail, but closer to the surface, then we can use the spatially offset technique. So I'll go to my conclusions. Monte Carlo modeling can give us information about how light is transported through turbid media. Microcalcifications are a good biomarker for cancer in breast tissue. And by looking at the different arrangements and geometries of lasers and detectors, we can look at different areas of the breast tissue and hopefully be able to bring that to a clinical use in the future. Thank you very much, and I'm happy to take any questions.
Okay, cool. We have 1 minute for question, if anyone have. I do have a question. So in the Monte Carlo simulation, did you consider it's just single scattering or multiple scattering? Did you consider the interference between the scattering?
No, we just treat each scatter as a isolated event.
Okay, I see. Thank you. If somebody else have a question, you can type in the chat, and yeah, Laura can reply in the chat.
Thank you very much.
Yeah, thank you. Next speaker will be Giuseppe. Am I pronouncing it right?
Yeah. Can you hear me?
Yeah.
Can you see my presentation? I guess it's gonna be in presentation mode in a bit, I guess. Yeah. So good morning from Milan. Thank you for Mr. Chairman for the introduction. And today we'd like to give a brief update on the recent results we got in this project, which consists in the integration of bioactive plasmonic materials in one-dimensional photonic crystals for optical detection of bacterial contaminants. So just to put a little bit of context, we would like to develop this kind of sensors for the the general aim of minimization of food wastage due to bacterial contamination. And so we aim at developing this kind of sensor for the integration in food packages or a smart food box. So the platform that we select to this job is our photonic crystal. I don't think I have to give much of introduction to this kind of materials, but I want to just say that photonic crystal cannot be regarded just as a passive optical components, but they can actually play, so can be active optical components. Indeed, if I mean anything that can, in principle, change the periodicity of the photonic crystal, this is just a simple case of one-dimensional photonic crystal, or the electric contracts between the constituent materials can, in principle, be regarded as an analyte. This is a beautiful example that many of you probably know, these are the morpho butterfly wings that are basically can be sensors of alcohols. So we started from this kind of concept in collaboration with Professor Stotonielli in Politecnico di Milano. So what we did, we started doing essentially was to integrate some plasmonic materials. In this case was indium tin oxide in one-dimensional photonic crystals, apply some kind of external stimuli that can be photodoping, or in this case is an electrodoping. And you basically, what you do is you act on the charge carrier density of the plasmonic materials. These has an effect, of course, on the plasmon frequency and on the complex dielectric function. So eventually you have an effect of the electric constant of the constituent material, and then on the photonic band gap. And then what you see is an active modulation of the photonic band. In this case, is a blue shift. So then we thought, okay, why we don't integrate some plasmonic material, which is not just a plasmonic material, but is known to be an effect on bacteria, for instance. And this is, of course, silver. Silver is known from millennia to have an important bioactivity. And what we, very simply, what we did first was just to put a thin layer of silver on top of one-dimensional photonic crystals. And what we did see is that when you put Escherichia coli, but we have data also on other bacterial strains, and depending on the kind of interaction between the bacterium and the silver, we do see different shift of the photonic band gap. Here is just a control experiment in which you see either the culture medium or the coli. You don't see anything, because this is just the photonic crystal without the metallic layer. So basically, the take-home message here was that the plasmonic layer is the responsive element of our photonic crystals. Then, after this first experiment, so we start focusing on the exact interaction between bacteria and silver, not from the biological point of view. Of course, here we have many reports on this kind of effect, but from the material point of view. So from the biological point of view, it's well known that the toxicant is basically silver ions, which are released by the skin, the oxidative dissolution skin, and then eventually these ions will interact with the plasma membrane. They lead to disruption of plasma membrane, production of reactive oxygen species, leading to the final bacterial eradication. But this is not the point. What we wanted to study is the transformation of the material upon interaction with bacteria. So we did a series of experiments, including both morphological, structure, and then our expertise, which is on optical experiments. And what we did see is that, so this is the granular structure, nanostructure of silver. Is not a continuous film, is nanostructured. So if you put the culture medium itself, it doesn't lead to any effect. But then when you put Escherichia coli, you see severe agglomeration of the particles at the close proximity of the plasma membrane. And then, so if you put the permealizer of the plasma membrane, this is a conjugate electrolyte synthesized by Guillermo Bazan in the UC Santa Barbara, you see that when you increase and enhance the release of silver ion and then the permeation of silver ion, you see a more dramatic effect. So more or less you have this three-dimensional network. And from the X-rays, you do see that when there is an interaction with bacteria, there is a strong amorphization, as indicated by the dramatic damping of the X-rays reflection peaks. From the optical point of view, the first thing we saw that with a very good statistics in different experiments, what we did see is a clear blue shift of the plasmon, which is enhanced again when you put the permealizer here. And then, so what we decide to do is that to gain more insight in this kind of effect by employing ultra-fast optical techniques. So this is an example of a pump probe experiment that we usually employ for getting insight into the excited state dynamics of materials. So very simply, you have two beams, the pumping which distributes excitation in your materials, the probe which monitor the transmissions of the excited states as a function of time. And for plasmonic material, you have usually two response. One is the interband and intraband response, and then there's the through the response. So you excite your material with your pump, and then your material will relax via three main mechanisms. One is electron-electron coupling, which is very fast in the femtosecond time regime. Then there is the electron-phonon coupling in the picosecond, and then longer there is the phonon-phonon scattering relaxation mechanism. Very briefly, what we did see, first of all, was that there is also in this case the blue shift, as indicated by the blue shift of the zero crossing peak in the transient absorption spectra. There is a very important enhancement of the electron-phonon scattering, so increase of the electron-phonon coupling. And very, very interestingly, there is a very clear damping of this coherent oscillation in the pristine silver or the silver treated with just the culture medium, so not contaminated. We do see this oscillation that we attribute to some coherent oscillations due to mechanical briefing of the thin films. And we have now a collaboration in this kind of on this topic. But then when you put the bacterium on top, you see that the damping, there is a complete damping of these oscillations. So we have three effects, and I conclude. The first effect, which is the blue shift of the plasmon resonance, then we attribute to the electron two main effects. So these are just some assumptions. Electron donation from nucleophilic agents that are present in the bacterial cell wall, so increase of the charge carrier density, and then there is a blue shift, or there is, and this is an important effect, as you can see, you could see from SEM images, you see that there is some important geometric effects, so agglomeration and reduction of aspect ratio. So there is an enhancement of electron-phonon coupling. So again, we assume that can be due to the increase of charge carrier density here, or to the decrease of the electron-electron lifetime, scattering lifetime, due to amortization. The damping, again, can be due to agglomeration and the increased inhomogeneous distribution of particles due to the presence of bacteria, and again to amortization. What we want to do, and where we are doing now, is to start from some materials of silver, which is either nanostructured or in which the distribution of particles is less wide. And then we are doing like more in-depth spectroscopy studies, for instance, using Raman spectroscopies. With these, I would like to thank, of course, all the people involved in this very interdisciplinary project, and Fondazione Colourful, which actually funding this kind of research, and again is focused on the development of photonic crystal for optical detection of bacterial contaminants. And of course, thank you for your attention.
Cool. Thank you for your talk. So we have 2 minutes for the question. Does anyone have a question?
Hi, Gushak. Hello.
Go ahead.
Hello.
Yeah, I can hear you.
My question is like, if you increase the concentration of this bacteria over this photonic crystal, what would be the effect? Like, I just want to know, like.
I mean, we have tried to do some dose-dependent effects, and down to, so usually we use a concentration of 0.5 OD, which means we are in the range of 10 million colonies. And if you go down to 1 order of magnitude, you still see the effect, but we haven't gone down to concentration in which we don't see anything yet. I mean, we don't have data on very, very low concentration. We've just tried few concentrations, so like spanning 1 order of magnitude, and we still do see an effect. So summarizing this is from 10 to the 5 or 10 to the 6, even 10 to the 7 colonies, you see this kind of blue shift, and then all this dramatic change in the morphology.
So why just bacteria? Like other things can also attach to these things, like silver.
So other
like
objects.
Other objects apart from bacteria, like some other virus. I can say some parasites, thing like that.
Yes, yeah. I mean, we have started working also with spores, with some spores and yeast as well. But I mean, I don't know other species. I'm not a microbiologist, but now we have a collaboration in which we study the optical change of silver and plasmonic photonic crystal with when they interact with yeast, with spores.
So I would like to ask that this, if you have, sorry to interrupt. Yeah, we're running out of time. Could you type your question in the chat so you guys can communicate more? Yeah. So now it's 8 a.m. London time, and here I am 3 p.m. Beijing time. So now I'm switching with Sebastian for the next session.
Thank you very much, Ming, and thanks for keeping the timing. So yes, indeed, we will move to the flash presentation. As we mentioned, timing is important. The reason being that at the end of the session, if we run out of time, we may be cut by our friends and colleagues who co-organized this conference to move to the concluding remarks, where we will give the prizes for the best talks by postdocs and PhD students or master students, as I understand. There would be also some runner-up and also a prize for the best flash presentation. So stay with us. After the session, you can move automatically to the main session, or otherwise at some point you will be moved automatically by our friends and colleagues. I suggest that we move directly to Alvaro with your flash presentation. I will share my screen up. Yep, it's coming. Yep. Alvaro, are you with us? I know that you are with us because I chatted with you just before. I cannot hear you. Oops. Otherwise, I will have to move to the next speaker. Alvaro. Okay, well, I will have to move to Wang.
Okay.
Okay, perfect.
Okay.
The floor is yours.
Okay, thank you. So, hey, I'm Wong from the University of Nonlinear, Malaysia, and over the next couple of minutes, I'm going to share my research with you. So, to begin, breast cancer has affected millions of lives around the world, but this number has actually dwindled over the years with the help of early detection. Currently, mammography and ultrasound are two of the most common methods for diagnosing breast cancer, but these are not always accessible nor affordable to everybody. And this is where my research comes in, as I have developed a streamlined approach to detect breast cancer that would just cost approximately 14 cents per test, require no expensive equipment, and produce results in just a few hours. This approach would measure the fluctuating levels of cancer biomarkers called microRNAs, or in this case, microRNA 155. I've done this by designing a nucleic acid system called hybridization chain reaction, or HCR, that uses only DNA molecules in the form of hairpins. In addition, I've integrated fluorogenic and label-free molecules called silver nanoclusters, or AGNCS for short, into the system to produce a quick two-color fluorescence readout. So when the blood samples are added, the microRNA 155 present will convert the DNA hairpins into DNA chains. And this structural translation will also affect the fluorescence profile from the silver nanoclusters, thereby turning the fluorescence color of the solution from red to yellow. So after establishing this concept, I've demonstrated that this tool can specifically detect microRNA 155 down to a single base difference in the entire sequence. And this platform works over a wide concentration range of microRNA 155 with a remarkable sub-femtomolar sensitivity. So in lieu of current breast cancer detectors, the proposed optical nanobiosensor offers a better alternative in terms of cost, time, and effort required. Likewise, this will ultimately lead to more effective programs, more people helped, and more lives saved. Yeah, thank you.
Perfect, perfect. If Alvaro is back, we can make his presentation at the end. We'll move to Alex.
Not yet.
Not yet? Oh, Alvaro, I can see you. You will be after Giselle if you are fine.
Okay, but can you hear me?
Yes, I can hear you. Alex is on now. Alex?
Okay. Hi, thank you.
Thanks
Thanks for coming to my presentation on the lens oscillator based on the photonic crystal model, or LOPC for short. It's a theoretical model that I've been working with for the last year and a half. And the sort of inspiration for the model was really in the iridoplasts, which Miguel and you were talking about yesterday. So for anyone who wasn't there, the idea is that in begonias, you have the specialized chloroplasts which have photonic crystal structure, and that's responsible for reflectance and possibly for absorption enhancement. The motivation for my basically simpler model is two things. One is that the structure has a lot of extraneous parameters from an optical perspective. So the thicknesses of the membrane and lumen are sort of biologically significant, but they're not really optically important. And another consideration is that because there are very low refractive index contrasts in the iridoplasts, very small changes to the thylakoid membrane refractive index that you've chosen can lead to quite large changes in the output of the model. So rather than try and get a better estimate of the thylakoid membrane refractive index, which is quite hard, I thought, okay, well, let's just take the most important feature from it, which is that it's localized absorption, and try and come up with, like, use the lens oscillators which are well understood and try and parameterize that. By switching to the simpler model, which is more easily parameterizable, we can ask questions instead, not like what does the iridoplast actually do, but what could be done with the submicron narrowly absorbing multilayer structure. And then that lets you ask questions like, what effects are there in these toy systems besides slow light? And what do optimized structures for reflectance or absorptance, say, look like? We'll be submitting a paper on that latter question soon, so keep an eye out for that on relevant archive websites.
Perfect. Thank you. Thank you to you. Giselle, this is your turn.
Cool, thank you. Hello, everyone. I hope you're having a good morning, and thank you for the introduction. As Sebastian said, my name is Giselle Rosetta. I'm a PhD student at Aberystwyth University. Today, I'm going to talk about polymer photonic crystals, which are known as polymer opals, and these are made of polymer composite nanoparticles. So we form bio-inspired films using rheological techniques, like on the left here, where I've got a diagram. Heated rollers melt shear the particles, resulting in structural order. And this is known as a method called bending-induced oscillatory shear, or BIOS. I've spoken a lot about it before. Like a lot of the structures that other speakers were discussing yesterday and today, we're examining a small refractive index contrast here of only around 0.12. So what makes our structures unique is that the iridescence is actually reversibly stretch tunable, as well as, of course, viewing angle dependent. So my research is looking at optimizing this BIOS shear process, and in these preliminary studies I've been examining several variables, and the resultant swatches of the photonic crystals I've made are shown here. So as you can see, we've got quite a decent range of blue-green coloration of a mix of intensities. The most important variable so far, we think, is the shear rate. So you can see from the top table, I think, that the slow shear films come out with a much stronger, much more visible iridescence than the others. And separately, I've used masses to add resistance to the films as they're sheared. And I hope that this is inducing higher shear forces. So we're looking at that, and I'm also looking at examining the process temperature, so the temperature of the rollers. In the future, I want to characterize these films in the same way as I've done previously, with color spaces, as I've discussed in the two papers that I've highlighted there. With these studies, we can do complete parameter analysis of the BIOS process for soft photonics manufacture and optimization, so hopefully our findings will carry over to other processes. Finally, for me, it's that time where I am searching for postdocs, so maybe you know of opportunities available at your institution, and if you have any funding available in photonics research, I'd love to know more. Thank you for listening.
Perfect, thank you, and good luck for your search for positions. We'll move back to Alvaro. We're back to you.
Yes.
The screen is yours.
Yeah, so my name is Alvaro Escobar, and I'm going to talk to you about structural color in gamma bacteria, because so far we only have seen a structural color in flower bacteria class, as we can see on the two images on the left side. But we discovered that gamma bacteria are also able to display a similar structural color as the flower, as we can see in the three picture on the right side. So one of the main things of the gamma protobacteria are that they are not gliding motility. So it's because it's the gliding motility what makes flower bacteria to are able to display this structural color. So what type of motility has gamma protobacteria to make structural color? We doesn't know yet, but we are start to looking into a few of them. So some Cangelas and some Marinobacter, for example, because Cangiella was discovered and described as a non-motile, but we think that it's really motile because we performed some time-lapse movies in the microscope, and we really can see that it's motile. And so when we also try to elucidate which type of motility. So first we check onto the flagella, trying to see if in a swimming essay was able to move as far as other species that we know that have flagella, but we were pretty sure about this thing. So we had a Marinobacter subterranean species that is from gamma protobacteria who lacks a flagella. So we compare the flagella knockout with the wild type Marinobacter subterranean, and we saw that the expansion of the colony was similar, and also the structural color and the also similar. So for now, we don't know what type of motility allowed gamma protobacteria to perform this nice structural color, but we will study that. Yeah, thank you.
Sebastian, you're muted yourself.
Sorry about that. Thank you. So yeah, I was just thanking Alvaro and moving to Svania. Thanks for understanding what wasn't clear, Svania. The floor is yours.
Thank you. I hope you can see my screen.
Yes, we can see your screen, and you can start.
And I guess you can hear me well.
Yes, perfect. Go ahead.
Okay. So hello, everyone, and thanks for joining me for my talk, which is entitled Hot and Shiny, Enhanced Photothermal Absorption in Iridescent Feathers. My name is Svana Rogala, and I'm doing currently a postdoc at the Biofísica Institute in Spain. And in my talk, I will present some research that I did during my PhD, which I performed at the Evolution and Optics of Nanostructures group at Ghent University. So as you might already guess from the title, my talk is going to be about birds. And we have already heard it multiple times during this very nice conference. Birds are very amazing because they are very diverse in color. And color in bird feathers is produced within structures that are made of keratin, such as the barb or the barbule. And it is produced by pigments or nanostructures. So the most common pigment in bird feathers is melanin, which can produce gray, brown, or black colors. This is followed by carotenoids, which usually produce orange, yellow, or red colors. And also colors can be produced by a combination of different pigments. So for example, a combination of melanin and carotenoids can produce olive colors. The absence of pigment often results in white feathers. And of course, there's also these very amazing iridescent colors that are very spectacular. And iridescence is produced by nanostructure arrangements of melanosomes. And when looking at these feathers from different viewing angles, we can perceive different colors. Different colors can serve different functions. For example, bright and shiny colors often serve for sexual signaling. On the other hand, more dull or more cryptic colors can serve as an agent of camouflage. However, different colors can also have different thermal properties. And this is because different colors differently absorb, reflect, and transmit light. This is well studied for dark and bright colors. For instance, it is well known that dark feathers absorb more light than white feathers. And this means that they would heat up more when exposed to a light source, for example, the sun. However, the thermal properties of different color producing mechanisms are not yet known. So to study the thermal properties of different color producing mechanisms, I looked at sunbirds, because sunbirds are very diverse in color. So you can see here in the slide, all different species of sunbirds I looked at, because they have all these different color producing mechanisms. So we have iridescent colors, so iridescent blue, green, purple. We have unstructured melanin-based colors such as brown, gray, or black. We have carotenoid-based colors such as yellow, orange, or red. Of course, there's also a combination of melanin and carotenoid, so there's olive colors, and there's also white. To study the photothermal properties of sunbird feather coloration, I first looked at the optical properties. Therefore, I went to the Field Museum in Chicago and looked at museum specimens of sunbirds, and I performed reflectance measurements on different color patches. And I show you here the reflectance spectra for the different color categories. So I show you the average reflectance. So for example, in blue, I show the average reflectance spectra for all iridescent color patches. In black, all the melanin colors, and then it's followed by olives, carotenoids, and whites. If we look at the reflectivity of these different color producing mechanisms, we found that iridescent feathers would actually reflect less light than unstructured melanin-based colors, than olives, than carotenoids, and white. I then followed up with studying the thermal properties of these different color producing mechanisms. And therefore, I exposed the specimens to a light source, which has a spectrum which is relatively similar to that of the sun. So for example, we have visible light, but we also have a part of the near infrared. And during 10 minutes of a heating experiment, I measured the surface temperature of the different color patches using a thermal camera. And as we expected, different colors would heat up differently. So here I show you some example color patches, where on the left you can see the different reflectance curves, and on the right you can see the surface temperature as a function of time. So we have the 10-minute heating experiment, and for the following analysis, I look at the asymptotic maximum temperature. So therefore, I look at the average of the last 100 seconds of the heating curve. And as I already mentioned, iridescent feathers reflect less light than unstructured melanin-based colors, than olives, carotenoids, and whites. And this is also reflected in the surface temperatures that we measured. So iridescent feathers would actually heat up more than unstructured melanin-based colors, and more than olives, carotenoids, and whites. Interestingly, in sunbirds, iridescence is produced by melanosome platelets within the feather barbule. So here I show you the malachite sunbird, Nectarinia famosa, which has iridescent green feathers. And you can see here nicely that the melanosomes are shaped as platelets that are arranged at the surface of the feather barbule. Non-iridescent melanin-based colors, such as black, are produced by melanosomes that are shaped as rods or spheres in sunbirds. So here I show you the amethyst sunbird, Chalcomitra amethystina, and you can clearly see that the melanosomes are shaped as rods. I was wondering whether the different melanosome shapes or arrangements can explain the higher heating rates of iridescent feathers. Therefore, I used numerical finite difference time domain modeling in collaboration with collaborators from the Akron University, so with Anbei Patel. And together we modeled the structures that we see in the feather barbules. So therefore we used models of keratin blocks with incorporated melanosomes of different shapes. And these were exposed to a light source, a plane wave light source. And so first of all, I looked at different optical and heat models of different melanosome shapes and arrangements. So we use melanosomes that can be found also in nature. So we have different shapes like spheres, ellipsoids, rods, and platelets. And we once modeled like an organized arrangement and a random arrangement. And as you can see here, the different shaped melanosomes, they increase in aspect ratio. So we can go from spheres to platelets. What is important to mention here is that these melanosomes that are in the block of keratin, they are, the content of melanin within the block is always the same. Okay, so let's come to the results. If we look at the different melanosome shapes, we can see that models that contain melanosomes that are shaped as platelets would absorb more light than, for example, the ones that contain spheres. So the absorption would increase with the melanosome aspect ratio. So here I show you the platelets in red for the organized structure, but we found a very similar result for the random arrangement. So even also here, the melanosomes shaped as platelets would heat up more. And if we look at the temperature, we can see the same effect. So the temperature would increase with melanosome aspect ratio. So platelets would heat up more than rods, than ellipsoids and spheres. However, if we look at the organized structure versus the random arrangement, we barely see a difference. Also, if we look at the temperatures and compare these temperatures that we modeled here with the ones that we measured in the experiments, we can see that the temperature increases were rather small compared to what we measured. So these temperature differences might not be sufficient to actually explain the differences that we found between the heating of iridescent and non-iridescent feathers. So this is why we had to dig a bit deeper into the topic. Interestingly, in sunbirds, the iridescent feathers, or also in many other iridescent birds, iridescent feathers contain greater melanin quantities than non-iridescent based colors. So you can see it again here in the malachite sunbird. We have very densely packed melanosome platelets, and you can see that it's quite a large quantity inside the feather bubble. So I was wondering whether the greater melanin quantities in iridescent feather bubbles can actually explain the heating rates of iridescent feathers compared to non-iridescent melanin-based feathers. And therefore I used optical and heat models of feather bubbles. So we used transmission electron microscopy, and here I show you the TEM images for different bubbles. So we have the white, gray, black, copper, green, and purple. And in the bottom row, you can see the models that I built based on these images. So we always have a keratin block, and we have different melanosomes inside that are shaped differently or arranged in different amount of layers, and also that might differ in thickness. Overall, we found that the modeled and the measured reflectance curves of the feather bubbles would give quite a good fit. So you can see it very nicely, especially in the iridescent peaks here, that the modeled and the measured reflectance, the peaks, they match. So we produce a right color with the models. What we also found was that we have a similar trend in the modeled and the experimental measured surface temperatures. So if you can see here, we have the white, gray, black, copper, purple, and green colors. So here, these last three are the iridescent colors, and we can see that these iridescent colors would actually heat up more. If we then look at the melanin content, we can actually see that the temperatures will clearly increase the melanin content within the feather bubbles. So here, when I talk about the melanin content, I talk about the total width of melanin layers. And you can see clearly that the iridescent green feather bubbles, which contains quite a lot of melanin, would also heat up most. So overall, the conclusion from this talk, we can say that iridescent feathers reflect less light and heat up more. And this is less likely because because the melanosomes are shaped as platelets, but it's more likely because they have greater melanin quantities within the iridescent feather bubbles. And my results, and also other results that where I still talk about spectral effects, like comparing near-infrared reflectance and visible reflectance, and also looking at the temperatures that actually reach the skin temperature. All this is written up in a very recently published paper, which is entitled Enhanced Photothermal Absorption in Iridescent Feathers, and which was published in the Royal Society of Interface. And therefore, I would like to thank all my collaborators, my co-authors, and of course also to the Living Light Conference Organization Committee, and of course to you. Thank you very much for listening, and yeah, I'm looking forward to receive any questions. Thank you very much.
Many thanks, Svania. There are some questions in the chat. Unfortunately, in the interest of time, I won't have the time to take them orally, but of course answer them in the chat, or you can move in a coffee room that we set up. We will have to move to the next speakers, which is Hilary. Hilary, you can share your PowerPoint with us. Okay, I can see your slide. I can't see you. I can't hear you.
There we go, sorry.
Okay, perfect. I can hear you, I can see you. You can
Here we go.
start whenever you are ready. Perfect.
Perfect. So yeah, I'm Hilary. I'm a PhD student at the University of Bristol, and I work with Heather Whitney and Tom Oliver, and closely with another student, Viola, who presented yesterday. And I'm going to talk to you today about a technique that we use to measure the relationship between photosynthesis and photonics, or that we're trying to. But before I go on to talk about the technique, I really need to touch on quickly to an element of photosynthesis. So photosynthesis is a hugely complicated process, but for the purposes of this, I just need to focus on the first step, which is light harvesting. So when light hits photosynthetic tissue, the photons can follow one of three pathways. They can be re-emitted as fluorescence or chlorophyll fluorescence. They can drive photochemical reactions involved in photosynthesis, or under excess light conditions, photoprotective mechanisms kick in called non-photochemical quenching. And these can essentially be thought of as the harmless dissipation of excess energy as heat. And these three pathways are in competition with each other, which is really handy because it means if we can measure one of them, we can infer information about the performance of the other processes. And this is the case, and this is the premise behind quite a lot of photosynthetic research, that we can measure chlorophyll fluorescence, and this allows us to calculate parameters surrounding other photosynthetic processes. So most chlorophyll fluorescence measurements are intensity-based, but I'm going to talk to you today about a time-based measurement. So time-correlated single photon counting, or TCSPC, can essentially be thought of as a really sophisticated photon stopwatch. I'm going to quickly walk you through how this works. I'm hoping you can see my cursor. I forgot to change it to a laser pointer. But basically TCSPC starts off with a really high power laser, and these are sending out ultra fast pulses. These are directed by a beam splitter to two different places. So initially to a first detector, which sends a signal to a capacitor to start ramping. Meanwhile, the second beam is directed to the sample, or a fluorophore, which then becomes excited when it takes the photon in, and after a period of time re-emits it. This then hits the second detector, which tells the capacitor to stop ramping. We then have a total capacitance, or a voltage, which can be converted into a time using a time to amplitude converter before this is processed computationally into a fluorescence lifetime. So these are single photon emission events, and TCSPC essentially accumulates thousands of these and bins them over each other in a histogram to give a fluorescence lifetime. These can be used for all sorts of things and are generally used for reaction dynamic studies, or probing solvent environments, or energy transfer studies. But as I mentioned earlier, we're using this to measure chlorophyll fluorescence, which can be used to infer information about photosynthetic processes. We can repeat these chlorophyll fluorescence lifetime measurements as well and see how they change over time with light exposure, so either in terms of intensity or wavelength. This sounds quite complicated compared to lots of the intensity-based measurements that some of you might be familiar with. And the reasons for doing this is it offers several advantages over these intensity-based measurements. So it's not prone to things like photo bleaching or chloroplast shielding. We can also get a really high resolution, so on the order of 100 picoseconds, which is actually determined by the speed of electronics, and isn't too far off these single photon emission events. What I'm going to talk about today and sort of applying this to is looking at the role of naturally occurring photonic organelle. And these are found in quite a wide range of species, found all across the phylogenetic tree. So we've got a higher plant called begonia, and a photonic chloroplast called iridoplast, which has been spoken about a few times over the conference. So in begonia, we're really interested in comparing these sort of photonic chloroplasts to regular epidermal chloroplasts found in the leaves to see if this ordered nature can offer any advantages. It's previously been shown using static intensity-based measurements that it can enhance photosynthesis between 5 or 10% in low light conditions, but we're hoping to perform dynamic measurements on this. Another species I'm really interested in is a spike moss called Slagenella, and through the genus there's another type of photonic chloroplast called a bosonoplast. I say photonic with my hands up because there's a certain level of order, but it's not clear whether it's actually completely photonic really. But what these bosonoplasts do present is a really interesting opportunity to compare this really ordered upper region, which I hope you can see I'm indicating to, with the more disordered typical lower region. And we can do this obviously in the same organelle, which takes out some other variables. The third species that I'm going to talk about today is Ericaria slaginoides. This is a brown algae, which is previously known as Cysticerca tamariscifolia. And it's not a chloroplast within the brown algae. They're opal-like photonic crystals, which are essentially sacs of monodispersed spherical lipid droplets, which are found in close proximity to the chloroplasts. And they undergo a reversible light-induced disordered expansion, where we see the expansion of this photonic lattice. And I should have mentioned earlier, of course, though I think it would seem obvious that the interesting blue and green colours we're seeing in these species are obviously a result of this photonic nature. So yeah, in the brown algae, I'm interested in comparing dark adapted and light adapted states to investigate the role of the photonic structures. So how am I going to do that with a TCSPC? And essentially the answer is that we've built and integrated a reflectance microscope with the setup. And this will essentially allow us to correlate reflectance measurements, which are sort of indicative of order and photonic structure, with the chlorophyll fluorescence lifetime measurements, which are indicative of photosynthetic processes or the performance of them. I've got the scheme of what we've built up here. It looks like quite a complex optics setup, so you can mostly ignore lots of it unless you're interested. I just quickly wanted to walk you through the different sources and detectors. So we've got over here on the right, the TCSPC laser. And is used forming these photos, and this is mostly for safety considerations. So these are then aligned through the optical setup, and once they've gone through the sample and the microscope, they come out in a split between various detectors. So the first I've signaled is the TCSPC detector. I've sort of grayed this out because we've not fully integrated this part yet. We've also got a spectrometer, which can quantify wavelength and changes in wavelength, as well as a camera, which we can use for reflectance imaging and working out where in the tissue we are. So as I said, we've not fully integrated the TCSPC part yet, so I can't show you any photosynthetic results, but I'm hoping in the next year or two that I will be able to. But for now, I'm going to show you the reflectance images and the spectra. So in species of begonia, not just in Grandis pavonina, which has been spoken about, but in other species, we've located the iridoplasts and taken spectra on these. In Slaginella, it's a bit less clear cut. Initially, I thought I'd located the bosonoplasts, but actually on closer inspection, depending on the Z focus, it starts looking suspiciously like a cell wall. And there's a bit of a complication here because in other species of Slaginella, we do have a photonic and iridescent cell wall. So I compared these to some of these as control species, and it does look suspiciously like some of them. So we're not sure yet whether the photonic region of the bosonoplast has actually formed. This has previously been shown to be a light-induced formation. So we're continuing to grow these under lower and lower light levels. I've got some confocal microscopy images of the original bosonoplast species, and you can see that there's sort of a blue sheet reflectance, I'm sure reflectance rather than fluorescence over the tissue, which I think does look, again, quite a lot like a cell wall, so sort of proving this. And last but not least, in Ericaria slaginoides, I've located the opal-like photonic crystals, and we're in the process of performing dynamic reflectance decay studies of these, and eventually we'll be performing sort of correlative time-resolved measurements of the two. And I've got over here on the right a TEM of this tissue. With that, yeah, thank you so much everyone for listening. If you want to ask any questions now or email me any, I'm not sure how much time we've got, feel free. I'd also like to thank Viola and Heather Whitney and Tom Oliver, our supervisors, and BCFN for the funding. Thanks so much.
Thank you, Hilary. If there is one quick question in the audience, I can take it, otherwise we'll have to move. Hello, Hilary. Thank you for the talk, says or wrote Miguel. What was the size of the spot measured? Was it bigger than the iridoplast, or did you focus only in the iridoplast?
Yeah, I forgot to mention that. So we have an iris sort of just after the microscope, and we do localize just on the iridoplast, and same with the bosonoplasts and the OPCs. So we're sort of focusing down, and this allows us to take like control measurements of dark and light measurements, so we can normalize this.
Okay, perfect. Thank you very much. We can move to Bram. I'm very happy to end up with a talk from Belgium. Bram, the floor is yours.
Yes, hi Sebastian.
Hi.
Thank you very much for sharing this. Can you see the slide?
Yes, I can see the slide. I can see you. I can hear you.
Okay, perfect. So indeed, I'm going to talk and indeed finish the conference here. That's always quite nice. About wolf spiders, wolf spider egg sacs, and how they thermoregulate. So within the lab, the EON lab at Ghent University, we mainly study color, how color are produced in animals, but a large part of our research, like Svana just told, is also related to the role of color in thermoregulation. So that's something I'm going to talk about specifically for wolf spiders and specifically for their egg sacs. Now, why these egg sacs, right? It's an interesting thing because wolf spiders, and almost all of them do that, right? They show a remarkable maternal care, right? So they're not like other spiders, they just leave their egg sac somewhere unprotected, but they really lay their egg sac, they construct it very carefully into a spherical object, and they carry it along with them on their spinnerets. They carry it along, of course, for protection, but they also show some incubation behavior. They orient it towards the sun. They actually also shift position, rotate it so that the top eggs and the lower eggs that are distributed within the egg sac get or apparently get the same amount of heat. And when they hatch, when the egg sac hatches, the juveniles get on the back, and you can see that on the picture on the lower right, get on the back of the female and hitch a ride for a couple of days, and every once in a while some of them jump off, so they also get a nice distributed dispersal. And every year with some students, we study these wolf spiders in the dunes here in Belgium, and we've been wondering if it can get really hot in these dunes, right? It could easily get 40, 45 degrees. And if you see a wolf spider running around with his egg sac and it suddenly hits a very hot patch of exposed sunlight, how does the spider know how hot the egg sac gets, right? Because it's crucial for their fitness, right? For their reproduction, that they're not overheating. So we were thinking, well, they carry around on the spinnerets, but that's probably not very sensory oriented, right? I mean, there's probably not a temperature sensor there. And also if they use spinnerets, they're always in the shade. So that's a different temperature they're measuring. So we're kind of thinking, what if they use the abdomen or their own temperature as some kind of proxy for the egg sac temperature? Well, the nice thing about that particular hypothesis is that we expect then a very straightforward hypothesis is that the egg sac temperature always has to remain below the temperature of the spider, because otherwise you cannot function as a proxy of egg sac temperature. And we've gone out and we took a lot of spider species. We have a bunch of them here in Flanders, and we also collected their egg sacs. So we took them, of course, when they were females, when they were having their egg sacs, and we see a huge variation, both in size but also in color. The lower ones are typically found near water. They're small and white. There's an entire range, although these are different species, right? There's an entire range of very brownish, beige, dark brown to greenish, even blue colored egg sacs, which is also interesting from a color production point of view. And we took a very straightforward approach, right? We put them underneath a heat lamp that was about or approximately the same watt or the intensity of a sunny day in the dunes, about 1,000 watt, and we looked at how they're heating up. So this is a bit shaky at first because I have to handheld it to get the spider into focus. We're looking at the back of the spider. So this is the cephalothorax, the head part. This is the abdomen to which the egg sac is attached. We've already put on the heat lamp. And you can see there's already differential heating, right? Legs heat up first, which makes sense. They're very small and very thin. But then already you see some differences. The cephalothorax is heating up more, right? So yellow, more red is hotter. And then the abdomen is actually slower, but the egg sac, consistent with our hypothesis, really stays behind in heating. It was very interesting to note. We see this in the field, we see this in all kinds of experiments. And this keeps going, right? So the spider is now about 38, while the egg sac is about 25, something like that. So there's a huge difference, which makes it interesting to see that it actually works. The moment it hits around 40, the spider starts to move, or this particular spider starts to move to a more shaded area, which also gives some more information on what their temperature preferences are. Right? But this is nice because we checked it in a live spider and we confirmed the egg sac is still continuing to be lower temperature. And we did this more consistently for a bunch of species and a bunch of individuals, where we put them on a petri dish and some styrofoam, and we just heat them up. And we can see a similar pattern also with dead animals, because these of course have been killed. So the top row is every time the spider, and the lower row is the egg sac. And you can see a similar heating happening, right, where spiders heat up first, the egg, especially the legs, right, and there's a huge lag, or at least a large lag in egg sac heating, which is consistent. I'll show the curves in just a bit. We also look at cooling, right? Just switching off or flipping off the heat lamp and see how fast they cool down. And it's interesting to see that at the cooling phase, the temperature of the spider and of the egg sac actually are way more similar, are a bit more converging. If you look at that from some more quantitative measures, right? So these are the same, these are examples of a bluish, a brownish, and a whitish egg sac. And you can see, right, on the x-axis time and on the y-axis temperature, so a typical heating and cooling curve, that the egg sac of all these three investigated species, but we've seen this for more than 100, I mean, not species, individuals of approximately 10 species. This is a very consistent pattern. The egg sac, which is always the upper graph or the upper line, is heating up faster compared to the egg sac. Interestingly, if you look between these species, and then of course the cooling goes really fast, right? The heat source is disabled, and they converge to a similar temperature, which also makes sense that they cool down in a similar way, because then you can still use your abdomen as a proxy. What is interesting to see is that the egg sac, regardless of size or color, seems to have a plateau temperature of around 35. So that's for all, even a bit more for the smaller whitish one, seemingly indicating that there's something going on there, right? They're different sizes, different color, but yet they have the same temperature or heating rate. Also for the spiders, the larger ones, for the blue and the brown, they get around 45, while the one which is smaller for the white egg sac gets around 40. So there are some differences as well. So we see and really observe in all these experiments consistently lower egg sac temperature. But how do they do it? And that's something we're gonna focus on, and also in the coming research. But we've also been doing some stuff already. And it's nice because it asks a lot of these evolutionary and mechanistic questions, right? So there's a bunch of wolf spider species. What we see from the 10 species we looked at is that it functions in a similar way, right? But that's interesting because these sometimes have very different ecologies. There's also, of course, other function of color that has to be taken into account. These bluish, brownish eggs that function as camouflage, but how do you relate that with thermoregulation? There's also different biomaterials, right? There's chitin in the abdomen and the body of the spider, while the egg sac consists of silk. But what we're now to investigate this question, how that they do it, are looking into is more at the color production itself. And we have some first ideas about it, right? So if we look at them with microspectral photometry, just to quantify the curves, the reflectance curves, they look really nice. I have to say, I really didn't expect them to be very colorful, almost intriguingly different than what you expect from when you look at it with the naked eye. But you can see that the brownish has a typical, almost pigmentary spectra with increasing across higher wavelengths, while the whitish one is almost a bit more metallic with way more individual peaks. And the bluish one, which is very interesting, has a clear peak around the blue area of 550. interestingly and directly related to our thermoregulation question is, of course, that we also looked at the near infrared, right? And there it seems that, which is responsible for absorbing almost 50% of the sunlight or the energy of the sunlight. And there we see that it's very high, right? So if you start with the whitish X-axis, that's just across the board, very high, in both in the visible and in the near infrared. But if you look at the brownish and the bluish one, they are quite much lower in the visible, which could be a camouflage thing. But then again, very high in the near infrared, which could explain why they heat up slower. So this is kind of almost like a mechanism that allows you to be darkly colored, but at the same time stay cool. Now, this is a bit of a stretch, right? But it's something we're investigating. We've been looking at SEM pictures on how these X-axes look like, right? So some of them are more or less coated, and that we know that they have a coating. But what's interesting is that for the bluish X-axis, and that also relates probably to the blue color production, is that the silk fibers seem to be more oriented in a single direction, at least more compared to the whitish one, which are a bit more crisscross and just differently oriented. Oh, sorry. If we look more into detail with the cross section, right, with TEM, this is kind of confirmed. There's differences in the sides of the wall of the X-axis, if you can say so, and there's also difference in anything that resembles like a pigment presence, but we don't know that. And again, confirms that, especially for the blue one, which could explain partly the color production, is that these silk threads face in a similar direction, and they also have a decreasing diameter. So we're looking further into that as well, if that could be an explanation for the high NIR or even for the blue coloration. But there probably is also a pigment involved here. So to conclude, we found that they remain consistently lower, the temperature of the X-axis, which is consistent with the hypothesis that the animal uses its own body temperature as a proxy. And we think, but that's something we're going to further investigate, that the high reflectance in the near infrared is partly playing a role in that. Thank you.
Thank you very much, Bram, for your presentation. Very interesting. I got quite a few questions. Hopefully, we'll have the time to answer some of the questions that I have, or mostly that the audience would have. We will see. I was surprised to see all your pictures at the beginning of your talk, to see that we had such spiders in Belgium. Whereas afterwards, I figured that the scale was very small, and these are, yeah, I saw such a spider yesterday, but you know, they are really tiny, but with your picture, they're massive.
They are very common. They are very common.
A real question now, on the following slides, you know, a bit further, I think that you make some measurements, yeah, here, and my feeling, yeah, here and before, yeah, the cooling and the heating, I think, the previous slide. You make measurements and it appears that these specimens are dead. How sure can you infer on the thermal properties after the death of the animal?
Yes, and that's exactly that's something we've been wondering about as well, right? Because we keep them for a certain amount of time in the freezer, which also dehydrates them, and that could be that the X-axis gets dehydrated less. But that's why we look at the living animals as well. And we see a very consistent pattern in also live animals in the lab and live animals. I checked 5 of them in the field in more natural conditions. And we see every time consistently the same pattern arising. The X-axis heats up slower than the animal. And that's exactly the reason why we did a couple of them while they were still alive.
Perfect. Thank you. Well, I see that it's already 49. I think it will be time to move to the closing remarks, to also get the prize, distribute the prices that I forgot to mention are offered by Biomimetics, published by MDPI, and also by Chemical Science, a journal that is published by the Royal Society of Chemistry. So I will invite you all to move to the main room, where we will have our closing remarks. Thank you very much for your presence. If we were in person, I would encourage you to upload all our speakers. But I will do this for all of you. Thank you very much. See you in the next room, in the other room, sorry. if you struggle, you have to hit the button leave the room at the bottom right of your screen. And then you will have to choose between leaving the meeting, don't click that, otherwise you may not get your prize, or leave breakout room. See you there?
Yet we are going to cut and upload as many of the talks as we are allowed to after your permissions forms that you have all said yes or no to, and we'll circulate those so that everyone gets the chance to see as many talks as they possibly can over the next few days and weeks. I'd like to thank you, everybody who's given a talk, whether that was a flash presentation or a 10-minute talk, and I'd also really like to thank the committee who put this together, because it was quite a lot of work, especially just trying to work out how on earth we could fit all of the different talks in and how we could host it, including the amazing graphics and all of the abstract booklet, which I'm sure you've had a look at, and all of that. So thank you very much to the committee for all of their hard work. And
perhaps you also deserve a huge round of applause for being the kind of mastermind behind all of this and instigating it in the first place. So it would not have happened without rocks. So I, for one, am very glad and grateful that you
Brenda, I mean,
started this and
it wouldn't happen without it. Great. And now I think we've only got one more thing to do, is to announce the prizes. But I don't know if they're still, I think some, we have a very a great algorithm which takes in all of the scoring which has been done. Each of the talks has been scored multiple times, and then all of the scores aggregated. And I think, is Mike here, or Mike is just,
I think he's still working on it,
checking the sums. Okay, right. Well, please do stick around. I think we're gonna play the Living Light playlist.
You can maybe mention the sponsors.
Oh, sorry, yes. The sponsors who provided the talks, who are, sorry, I can't see them anymore.
So there is biomimetics that is published by MDPI. We will offer the prizes for the best postdocs and PhD students or master students presentations. And chemical science, published by the Royal Society of Chemistry, which provide some voucher to buy some books in the catalog of the Royal Society of Chemistry. And that would be for the runner-ups as well as for the best flash presentation. And we thank them for this generosity.
This is the Living Light playlist, which we'll just carry on until we get Mike with the exciting news about the prizes. In a matter of minutes, please hang around and it will be very exciting, and then we'll finish the conference all together. I think that we have an answer. We have our prizes, our prize winners decided. Have we got Mike to announce them? Great, Mike.
All right, let me share my screen here real quick. So can you guys see my talk? You should be able to now. All right, it was really tough to pick a winner there. There was a lot of really great talks, and we had to do some basically coin toss tiebreakers at the end. But really high-ranking talks for all of them. We multiple members of the committee graded each talk during the sessions. We looked at the scientific merit, the structure, the visuals, and the presenting. And we came up with the score, and we averaged that, and that's how we got our prizes, our choices. Really like to thank our sponsors, Biomimetics and Chemical Science, for the prizes that the winners will get. We have best talk prizes for graduate students and for postdocs. Those are going to be 200 Swiss francs each. And then we have 100 British pound book vouchers for the best flash presentation, as well as the first runners up for best talk and yeah, and the first and second runners up for the best talk, one PhD and one postdoc student. And so here are the winners. The best talk from the graduate students goes to Avalon Owens. The best talk for postdocs goes to James Foster. And then the best flash presentation goes to Yasmin Eratini. The two runners up are Aubrey Miller and Benito Wainwright. We'll be sending you guys congratulations to all the winners, and thanks everybody for all the wonderful talks. We'll be sending a certificate to the talk winners, and we'll contact you about receiving your prizes after the meeting. And I think thank you to everybody for attending. This was really, really amazing and inspiring. And thanks to all the other organizers, especially Rox, who organized this meeting. It was really, really spectacular. And I think that will conclude. I have to give a lecture in 5 minutes, so definitely will conclude. But thank you all again, and have a great day.
Thanks, Mike. Thanks, everyone. Awesome talks, awesome job, so much cool science. Thanks for joining us from all over the world.
Thank you in behalf of the speakers, also for all the organizers. And just my personal note, I'm going to go to the breakout room number 3 and have a Tulam or Dew.
I would join Pramosh, but I have to work. Goodbye, everyone.
Bye, Mike.
Bye, Mike.
It is
Bye.
2 in the morning, so I'm going to go to bed. Goodbye, everyone.
Bye.